A very good morning to all of you. I, Dr. Jaspreet Kaur, welcome everybody here in this virtual training program. We, on behalf of Organizing Committee of Maharishi Markandeshwar, deemed to be university, extend a warm welcome to distinguished speakers, participants from different corners of the country, and dear students in this virtual 3 days program of Pharmacy Practice Module-VIII for healthcare professionals on Advanced Practice Internship. This training program will provide the knowledge of daily hospital activities like patient case studies, dissemination of drug-related information, detection and management of ATR, and information about skills and attitude required to provide patient care with special emphasis on rational use of medicines. Before we commence this program, we would like to bow our heads in front of Goddess Saraswati for her blessings and light of knowledge to us. Now, I would like to invite the guiding force behind this event, Dr. Sumit Gupta, Dean and Principal, College of Pharmacy, to say few words. Sir, please.
Thank, thank you, Dr. Jaspreet. Very good morning to all of you. I, Dr. Sumit Gupta, Chairman of this webinar, welcome you all in on this Pharmacy Module 8 on Advanced Learning Series associated with SPR. The objective of this program is to enlighten the healthcare professionals, especially PharmD interns, about hospital activities with specialized emphasize rational use of medicines. It is a great honor to us of official welcome our honorable speakers, Dr. BIKAS MEDHI, PGI, and Dr. DEEPIKA from NIPER, who accept our invitation and agree to brush up the things during this COVID. So I wish to organizing committee for this grand success and have a great day. Thank you, Dr. Smith.
We hope all of you will deliberate in this training program with full zeal and enthusiasm. To introduce our first speaker, Dr. Bikas Medhi, is really a great privilege for us. He is a dynamic personality with diversified knowledge. Dr. Bikas Medhi is a professor and additional medical superintendent in PGI, Department of Pharmacology. He is chief editor in Indian Journal of Pharmacology. He is expertise in regulatory, experimental, and clinical pharmacology. He has more than 20 years of teaching and research experience and have won more than 50 prestigious national and international awards. He is editorial board member for several renowned journals. He has authored he has authored 5 books and 60 chapters and published more than 200 articles in national and international journals. With long list of glory and achievements, I would like to invite Dr. Bikash Medhi to deliver the first talk of the day. Sir, please, Professor Bikash Medhi.
Thank you. Can you hear me? That audio is clear. Ma'am, audio is clear, and you can see the slide.
Yes. Yes, sir. Your slide is visible.
Okay. So we are going to discuss this very important topic, which commonly people discuss officially, unofficially. So this is a topic is relevant to all the society and all the stakeholders. So I'm going to talk about some of the crucial facts about quality of medicine in Indian scenario and also the global scenario, and how you can overcome the battle against the counterfeit drugs. So before I talk about it, you know that how many medicines are available at present in the therapeutic area. Many of us, we believe that we have medicine for all the disease. So rightly, if you see globally, we have more than 1 lakh formulation is available of 500 ingredients. But when you talk about the accessibility of medicine, half of the population, they are not, medicines are not accessible, especially the essential drug. And if you look at the budget, 35% of the health budget is spent in the, in the health sectors. And worldwide, if you see the budget is more than 600 billion budget. So earlier it was expected that in 2000 till 2017 it will go up to $1,600 billion. So this is a scenario that many of us, we talk about the medicine, that all the medicines are available for all diseases, but we can have number of diseases where not a single medicine is available. For example, in North Belt, we have a lot of select patients. We do not have any medicine. Like autism, we do not have medicine. So you can take number of diseases where rare cancers, we do not have medicine. So there is a scope to look for it. Now, when you talk about national health system, there are several major challenges. You can see the challenges have come in pandemic also, that we're looking for a new drug, we're looking for a new vaccines. So in order to provide that health product of the service, which is reasonable or reliable, or we want an efficient manner that all the medicine should be available to majority of population. Last year, we had conducted one of this program like this in PhD chamber, where the organizer was the owner of the Ketan fan. So he asked a question to the public: if the Bikaner Puja is available in the rural India, why not the medicine is available? So moral of the story is that we have to make a system where all the medicine should be available to all the sectors in inaccessible area also. So that is why the concept was started that we should have an essential drug medicine list throughout the countries. Every state, every hospital, they should have a hospital formulary based on the essential medical list. So this concept was started in 1977 with the publication of World Health Organization, that model list for essential medicine. So idea is that if you make a list, and based on the list, you should make it available, affordable, and it is accessible. Since this conception was started in 1997, the essential list was prepared, medicines which are utmost importance and are basic, indispensable, or necessary for health need for a population. And when you look at the data in 2002, it was prepared in order to satisfy the priority of the healthcare need for the population. So basically, when you select all the medicine, they are selected based on what are the relevance to level of evidence in terms of that it is efficacious, it is safe, and comparatively it is affordable. So WHO also had done a survey of almost half of the globe to 134 countries about national essential medical list, and majority have been updated because they wanted to implement this concept throughout the globe. So number of medicine that was included at that time, it was 397. So it is ranges vary from 334 to 580 medicine based on place to place and based on the disease prevalence and incidence. Now, coming to one provision of medicine, who are the people are responsible? Because you know that you can say that this is a group where because of them it is not available, but it is a responsibility of all the stakeholders. So you talk about ideal situation, then you talk about who are the people are responsible for, what are the self-interest. If you look at the group interest, that industry is manufacturer, then the regulator's responsibility is to provide the public safety. Then you talk about the doctor, physician, there is number of pressure over the physician that they need to prescribe that, most beneficial medicine. Then what about, other people like clinical pharmacologists or pharmacists, that we encourage that whatever treatment is given, it should be rational treatment. Then you talk about health economists, they talk about that pricing, how to maintain the pricing. We have a system for, controlling the pricing. So there are diverse group. Ultimately, that how you can make it available and accessible and affordable. So availability, when you concern at availability, because why I'm pointing out this, this are a point, because ultimately when you talk about quality, so you have to see that what are the infrastructure is going on and how do you improve it. So here if you see the interested party is MNC, multinational company, they have a legislator and it is regulated by drug controller, but drug controller do not have an authority to, in India to regulate the price. So it is by Minister of Chemical and Fertilizer, they have a drug pricing authority who regulate it. So earlier, when you make an essential drug list, and when it comes to the name in the essential drug list, the price is already controlled, or in case of emergency drug. So then another is those who are patent, so they can you cannot have a pricing control over the patent. So availability of medicine, it's a government responsibility of major purchase, because one example you can see that when number of drug has been included in essential drug list, when the pricing was controlled, then stop manufacturing. So all the essential requisites are in order to save, whether it is safe, efficacy, but at the same time, we have to maintain that a good quality manufacturer. That means whatever drug you are going to use in a human, it has to be manufacturing in good manufacturing capacity. So when you talk about GMP, there are two types of GMP we use in a domestic market. We use in a we export, where we maintain the WHO GMP facilities. So otherwise, we follow the Schedule M. So essentially, better existing medicine for positive cost-benefit ratio. These are so many factors you need to look for. So when you apply the different, stringent activity in various countries, like you also see that, like today, you take an example of why all the people who are coming to India for vaccine, because the capacity for larger population, it's only India has that capacity for medical. That is why Oxford has come. Other, Russia had approached India, because we have the largest facilities to manufacture. So local drug manufacturing capacity, that you talk about process patent or copies of innovator drug within 1 to 2 years. So we have an Indian policy, or parallel import. So these are factors are very, very important in order to make it available of medicine for a population. And earlier you can see that we used to import a lot of bulk drug from the China, and then we prepare the product and we export it. Now, in continuation of availability of accessibility of medicine, because we want to improve the health sector, depend upon the optimal provision of medicine. So again, the three factor is very important. That is it accessible, or is it acceptable, or can you afford it? So the policy and practice by a pharmaceutical company, or in relation to government policy, confirm the local situation that has to be implemented, and provision for medicine for affordable cost. This is very important, as you know that when you fix the price, that company has they have opportunity to increase 10%. So depending on the case, it has to be also be regulated. Now, just take a look at current list of the medicine available by Government of India. And if you look at that 2011, when we started, we had 348 medicine out of 653 formulation. Out of that, you can see the 47 drug is included in previous list, and that has been removed. 43 new drug been added in the current list, and 14 HIV or AIDS and 33 oncology medicine have been added. So tomorrow, it is an ongoing process. So we might include some of the medicine which is required for pandemic situation. So India at that time did not have any separate list for children, but that organization like Indian Academy of Pediatrics, they have published essential medicine for pediatric population, and they had included 134 medicine. Now, when you compare with WHO and India, you can see in India we have a unique, characterization that we have a lot of fixed dose combination. But otherwise, if you look at it globally, they only encourage that fixed dose combination FDC you typically call for tuberculosis, malaria, HIV, now hypertension. Already talked about leprosy, but we have maybe more than 1 lakh fixed dose combination that also been screened to eradicate all the irrational fixed dose combination. So you can see the comparison of WHO in terms of FDC, or which other drug is not included, various categories like sensory organ, respiratory system. You can see the comparison of WHO and India. But it is important that we only include the drug looking at Indian situation. That has to be looked at. Now, second thing is when you discuss about availability, accessibility, and affordability, available and affordability is a key component, and which is equitable to access of essential medicine. So that means we are looking for what are the requirements for Indian population. 50 to 80% is limited to access the essential medicine. So there is a study which is conducted in 36 developing nations or middle income group countries, which shows that there is a poor availability to generic or public sector. I think last 2-3 years we have been debating that should we prescribe the generic or should we prescribe the branded. and it has been also been approved, advised by the highest court, that preferably you should write in generic by council also. The doctors is going to write the generic name, and that is the regulation now. So WHO also reported that 1/3 of the world population is lack of reliable access of required medicine, and the situation even worse in developing countries. So this is still a practical challenge, that can you make it available in the most remote area. So second question is poverty or unavailability of essential medicine for more than 18 million dead, that result. So that has to be also be counted. So one thing is that medicine cost is not always a constant for affordability to the society, but at times you see the drug cost is around 25 to 66% of the health budget, especially in developing countries, and equal to 20% health budget for industrial nations. So despite of this having lower prices medicine, when you compare, give some of the slide that how affordable medicine in India compared to any other countries, that also you can see. Now you take an example of a very common drug we use, ranitidine or aspirin. So if you look at the price, prices India and Nepal is less than 2 rupees. But when you go to Pakistan, that same ranitidine is costing 21 rupees. You go to Korea, it is 61 rupees. Zambia, 82 rupees. So there is a huge variation. Or you go to South Africa, same 2 rupees price is 116 rupees. So these are the price like common drug like diclofenac sodium, ciprofloxacin, or acyclovir. You can see the difference of price. Moment you go to Western nation, then price is 3 times, 10 times, or even 15 times is higher. So it is 1:58 when you compare with South Africa or any African countries also. So these are the example that how the prices varies, and still we can proudly say that India, it is cost is much lower, but still we have to control the prices as per our national pricing authority. Now let's take a situation of developing countries or optimal drug policy, that how we prepare. Like we have a basic drug list, or we have an optimal drug list. We always check for efficacy and safety on new chemical entity. So normally that pricing controls, the legal provision is that compulsory licensing, parallel import. So we have a loan licensing also, that also many of these MNC or bigger companies, they manufacture in a very smaller, or then they market it, and then there is challenge in improving distribution and storage facilities. And these are things are there. Of course, you can see some of the example that how NIC that they're coming with a software that if you buy medicine, it is manufacturing, suppose in Bati, it is the largest one, and how it distributed, you get it from the software, and how much it is sold in different states. So that's provision is also going to implement for this. Now coming to the crucial question is how much we spend for health sector. Now you take an example of total expenditure of the GDP US, they spend around 18% in 2010, and you talk about small countries like Timor or Maldives, they spend 6%. Now India, we spend 4%. Now second question is, over a period of time, last 10 years, how much has been increased? So you take an example. In 1995, in US, they used to spend 13.6%, and after 10 years, they have increases to 16%. Like for example, if you take an example of India, India used to spend in 1995 4.1%, but after 10 years also it is almost a static one, 4.2%. So that is a situation that if you look at the different state also in India, expenditure state-wise, highest is spent by money has been utilized by Kerala, and the lowest is by Bihar. So you can take an example that how many countries. So you can see that how much money has been spent in health sector in Kerala, and you take example of Bihar is the lowest one. So this variation is also need to be uniform, that how you can spend, or a systematic wise, that you can have practical implication that there should be output in health sector. Now another thing is that the concept of New India, like some of the state is much doing better. You take an example, when you go to a WHO meeting, you get to know that Tamil Nadu is the first state to develop, essential medicine or looking at the quality. And second thing is, referring to Dr. Ranjito Chaudhary, who come with the drug policy during the initial time in Delhi with a RUD program. So second state is Rajasthan is also doing good. So first essential medicine was prepared in 1996. List was neither implemented or nor procure of medicines for drawn up. Then first and second version was published in 2003 and 11 respectively, and later on it come into implementation in a terms of price control also. That moment the drug is incorporated national essential list, that the price is going to regulate. So there are huge disadvantages or huge advantage for this selection of limited. So basically, if you see that we wanted to make it available a good quality medicine. So that is why that essential medicine list does not give information for funding or purchasing, but it is also that it is given that based on that you can procure the medicine and you can check for good quality of medicine. So one of the weakness may be that people may not stop manufacturing. Then the question of affordability. You can take an example of snake venom. You can take an example, number of example you can take it, that where that manufacturer will not be interested that if the price is controlled too low. So that also need to be looked at. So these are some weakness. I'll not go into detail. But however, the Indian perspective is currently you are making it like earlier used to say drug, then you say medicine. Now we used to call it product because there are a lot of materials that are also available. So this has been updated, and we encourage the policy of RUD, National Use of Drug, that I have referred to Dr. Ranjito Chaudhary, the Delhi Society for Promotion of Rational Use of Drug. That is what the implementation in Delhi initially with the base of recommendation from Dr. Ranjito Chaudhary that you can pool the medicine and you can check the good quality of medicine. Then if you know that requirement for a whole year in all Delhi, or similarly for all the state all in India also. So India, this survey about, regularly we do it. I'll talk about the survey, how it has been done. WHO also published some of the list of essential reproductive health. So this need to be looked at. Now, as I said, that drug price control, that is by Ministry of Chemical and Fertilizers. So we have a regulation that how the price is controlled. So in conclusion, if you see that medicine, how we procure, that the concept is that some of the medicines are not available, especially in the rural or remote area, that need to be make it a point that all the medicines should be available for this. Now, there's a huge concern about dietary product also. Now we have a FSSI, and if you see that nutraceutical or dietary supplement, this is another issue with contamination and quality is not mentioned. Recently, we had an issue with artificial sweeteners where we commonly use. Some of the sweeteners are available in the markets, which if you put in the hot waters, it releases methanol. So that is a concern for pregnant women or in children also. So then how do you regulate all these activities like advertisement? If you see, you take this dietary supplement, take the drug, and or in terms of obesity, these are exaggerated ones, so it need to be regulated. So ultimately, this is one sector of maintaining the quality. Also, dietary supplement or any dietary product has to be there. Another sector is that this dietary supplement has been used tremendously in all the games and players. They are very popular because they wanted to boost up, get a medal. So there is a huge list that India become, number 2 and number 3 in doping also. This is another concern that if you control the quality, then it is going to come back this violation of anti-doping rule also. Now, third sector is, in order to maintain the quality, we have a PPPA program where we maintain all the records. All these, the problem with the qualities or whatever the adverse effect with this, the medicine, we always spontaneously, or targeted spontaneously, we collect the information and we record it. So these are the database, and the regulatory is involved for that. I'll not go in detail. So we have a program right now. We have 4 lakhs database. So this is what and quality. In terms of reporting of safety data, so we are at par with US. It is now 0.94. So this is what the database is growing. So we are going to take the importance or that how to extract this information in order to have good quality data. India is contributing right now almost more than 2% of the global database. Some of the issue I'd like to talk about is recently there's a recommendation that there's EPharmacy. So from PGI, we had conducted one of the preliminary studies that what about behavior? Because when I attended a couple of meetings in Europe, we could find that they were saying that there is a bad quality medicines which is coming from a place like China through Hong Kong, or maybe some portion from India, very limited, or from Brazil. So that is what, in case of e-pharmacy, how the population actually look at that in terms of quality, or whether they have awareness or not. So we wanted to evaluate the knowledge, attitude, and practice of Indian population toward e-pharmacy. So it was a prospective cross-sectional studies, and population was aware that there are further evaluation of the knowledge. Even if you ask a person who is buying the medicine from pharmacy, that at least they know that they ask for expiry to nahi hua. So this is very important point, that how do you evaluate. So the result of the study shows that 83%, almost more than 80% are aware of online pharmacy, but only 25% they use online pharmacy. The reason behind, they are familiar with buying the medicine from the offline. So, but they do not know that in case of a prescription drug, or cosmetic, or dietary supplement, these are mostly 27% prescription drug, or cosmetic is 22%, or supplementation. And there are huge number of people who buy the herbal medicine also, and non-prescription drug is 6.7%. So this is what, despite of the awareness regarding online pharmacy, it is yet to people prefer to buy offline, and which may be due to the perceived of possible risks to the poor quality of medicine and lack of trustworthy. Now, coming to the exact, the topic, how you assess the quality. What are the government regulations? What do you mean by moment you say quality of medicine? What does it mean? You say NSQ. NSQ is not of standard quality, or you say counterfeit medicine, or you say spurious. So there are three words used: quality of medicine is not of standard quality. That means whenever we market any product, we always state the standard at par with Indian Pharmacopoeia. So in India, whenever you are going to that, it is at par with Indian Pharmacopoeia, or you happen to go to US, at par with USP, United States Pharmacopoeia, or BP in UK, British Pharmacopoeia. But if somebody come and say that my product is at par with USP or BP, it is acceptable. So here, the regulatory word NSQ use accepted, spurious is accepted, but counterfeit word is still not included in regulation, because many of the products are very, very popular. So they are marketing, like you take an example of Bata. Everybody knows of Bata. So there are a company called Rata also, which is people confused with Bata, they buy it. So finally you come to know it's not Bata, it's Rata. Similarly for medicine also it is like that. So there are many medicines which are marketed in a popular brand, but exactly it is not marketed by. So this is what the example of counterfeit is also. So I'll talk about the infrastructure, how do you test the drug, and how do you function with the testing laboratory, how do you maintain the quality trend every year. And as I already told you that India has conducted largest survey initially with PDSET. He had conducted a survey for quality of medicine. Then it was done by CDSU. Then Ministry of Health has largest survey where 48,000 samples were collected across 676 districts of the remote places, and it was assessed or 1586 classification of the drug. So you can see a survey report. What about the scenario in India? We will get a clear picture for them. So as I said, the quality of medicine is essential for efficient disease management, because if the quality is not good, then even if you prescribe, like we have a situation of antimicrobial resistance, like for example, that question is why TB is increasing, why malaria is increasing. So then you can see that if you are giving the drug, because the reason behind, if you talk about regulatory today also, is just referred to Second World War, where in Indian Army were under British were fighting in Burma area. So during that time, if you see that there is a concern of use of quinine, because quinine was supplied from the UK, and after giving quinine also, that army were not getting relief, they were dying of malaria. So then later on, the group of people, they launched a complaint and in terms of PIL to Bombay High Court, and that's how Indian regulatory system was established. And there are two tier of Indian system of regulatory, that is central regulatory system and state regulatory. Every state has a drug controller office. So this is to make it that regulator, whatever product they approve to the market, it's a good quality. So you talk about pharmaceutical analysis of important the quality of active pharmaceutical ingredient like API and dosage form. Then we have several technique for identify the quality, identify the purity and strength of medicine by quantitative and qualitative. And regulatory authority makes sure that this is through this mechanism, batch to batch. Even in a hospital, when you procure a medicine, you should have a system of randomly taking this batch and send for analysis to any of the government lab. So that should be the mechanism in order to prove that though the company, the manufacturer give a certificate, this is a good quality, but you should randomly also do it. So the quality is affected from improper storage. This is very important. That is why we have to put more of this importance, that how you distribution phase, how do you maintain the storage condition, especially in vaccine. But you know that we have extreme weather. Summer it might go to 45, winter it is also very cold. So during the extreme weather, you have to keep that distribution phase. Now taking the sample and testing of the medicine from different point also very important. And it should maintain a higher standard of the medicine. Otherwise, you'll not get the exact current scenario. So that is why the drug survey was done to see the scenario in India and how should we go in future. So as I said that we have conducted almost 3 surveys by BDSAID, then by CDSU, then larger survey by Ministry in 2014 and 16 onward. So this is a major survey was done. So not all the countries of stringent system of medicine regulation is in place, but that is why that India has a system that, as I said, we have a 2-tier regulatory system. One is central regulatory system. It is called DCGI, Drug Controller General of India. Every state, every UT, they have a drug inspectors and the drug controller. So this is to make that quality of the medicine in the market should be at par with any of the pharmacopoeia, Indian Pharmacopoeia or USP or British Pharmacopoeia. So these all to be done in order to make sure that quality is okay. Then you authorize, and based on this quality of the survey, depend on accuracy, reliability, and interpretation of the data. So there are some of the publications which say that WHO used to have draft norms for survey for assess drug quality. There is a lot of allegation also. So, but if you see that government data, that our not of standard quality is around 3%. So that need to improve. Now, those who are student, when you are posted in the lab, and you should know how to do it. So I'll give it a background also that all these labs should be accredited lab, and at least you said that when the report goes, and then you can say that these are done in accredited lab, and that is why sample quality compliance that we during that time when we done the survey, we had a plan A, and how you collect a sample. We had developed a software called AKS software. Then sample were received in the lab by registered post. When you collect it from the outlet pharmacy, then complete analysis is done as per the Pharmacopoeia standard, and review. It was reviewed by a committee. Then only it was compiled with the survey. So the focus is on basically the quality, that how you maintain the quality. So these are the example that how we procure the medicine for different outlet, like we put it as urban area or even the import and export. Like in the airport, we collected a sample, or you take an example of shipping area where sample were collected, because we wanted to see that is there any problem when we import the medicine, or is there any, what is the quality in export of medicine. Then it was collected, then it was stored in NIB, that is National Institute of Biologics. It was a coordinating centers, and all the RTDL lab or CDL lab, it was done analysis, and that's how the report was prepared. So there is a big survey was done, as I said earlier, that extent of problem of spurious and not of standard quality medicine. It was started in 2014. Now, when you talk about how you scientifically collect the sample in order to evaluate the NSQ or dosage form. So you can say that there are, as I say, 15 ATC classification drug. So you collect the tablet, you collect the capsule or parenteral preparation. So total number of collection, sample analyze, number of sample standard quality, number of sample of standard quality, and how many are out of them are spurious percentage wise, or how many are not of standard quality. So this was analyzed through this form. So then source of collection like medical store, government hospital, ARC, what I say that import and export. So everything, it was collected from different point of view. Now, many of us, we believe that medicine available in a remote place like rural area, it may not be a good quality. So that is why we divided into India in all the district. We make it a point that every district we are going to collect from the village, and small cities, or you take an example of metro. So every different region it was collected. Now, when you talk about quality, it is important field that most important medicine or it involved. As you know that when we manufacture a medicine, we make it a point that we follow the good manufacturing practice. And during the development also, we follow good laboratory practice. And at the same time, that in the process, we maintain the quality assurance and quality control. So any lab you go, that how do you do? So they have mechanism of internal accreditation, internal quality management, and there is a external quality management also. That means internal quality, they have a QC. So there is a team who look after all the procedure, and that is why they maintain the quality assurance and quality control. So basically, quality control, when you talk about, it is a critical process for analysis or carried out the drug testing laboratory. So as I said, that it is standard is maintained in terms of we talk about Indian Pharmacopoeia or British Pharmacopoeia, or you talk about USP. So globally, it is accepted. So here, the series of qualitative and quantitative tests is done. Like we use some of the instrument like SPLC, FTR, spectrophotometric, depending on that medicine. And we have an SOP for doing that criteria for how do you standardize or how do you make sure that this is substandard quality or a spurious one. So there is a method for doing that. So this is basically a multi-level testing that you take the medicine from the API or any of the outlet, then you take it to formulation, then ultimately you check for the quality by this method, step by step. Now, let's discuss about the Indian infrastructure of the government supporting infrastructure. Like we have many labs, like we have a central drug testing lab, that is the Apex lab in Kolkata. But every state also they have a testing lab. And we have also private drug testing labs. So you happen to go to Panchkula area. So there are several private testing labs also there. So here you have to see that when you send for a testing lab, then you have to also ask this, is this lab accredited lab? So just take an example, as I say that Civil Kolkata is a Apex lab applied. So you remember that situation where somewhere in Chandigarh, there is an issue of ciprofloxacin. So sample was sent to Kolkata lab. Similarly, then we have a IPC, Indian Pharmacopoeia Commission, that is in Ghaziabad. Then we have a lab in NIB Noida. So that is, this lab is particularly for all the biologics, like we talk about insulin or biosimilar, it goes to National Institute of Biologics in Noida. Then we have CDL, especially for vaccine. We have a central drug laboratory in Kosoli, CDL. Then we talk about RTDL in Chandigarh. It is regional drug testing lab in Chandigarh. Then one in Guwahati, regional drug testing lab is in Chandigarh. Then central drug laboratory in Bombay. Then we have in Hyderabad, central drug laboratory. We have in Chennai. So altogether, if you see the infrastructure of central government, we have total 9 labs. So one is Kolkata, then Hyderabad, then Chennai, then Bombay, then Noida, IPC and NIB. And all are RTDL. Now, so their responsibility that because every drug inspector that we have more than 2,000 drug inspector by far now in 2020. So all the drug inspector, what it does, they goes to the different outlet. They have to collect in a month 25 sample. All these 25 sample, it goes to it is depending on that whose lab they would like to send it to. So they can send it to Kolkata or any other lab. So their responsibility is to collect the samples from the outlet, retail outlet, then send for analysis. So this is a continuous mechanism in order to maintain a standard of medicine. So responsibility of the lab is they have to analyze as per Drug and Cosmetic Act 1940 and Rule 1945. Then we have a rule for medical device also. Then for vaccine, biologics, all the recombinant products. So the all central drug testing lab you talk about is around 18,000. So as I said, that number of product they collect is 25 sample per person. So the number of sample declared on an average, if you see, take an example of 2013 and 14 is 560 by CDL. So it is alert is done by monthly wise. Now, just look at the infrastructure of test that every state there is a drug testing lab. So the
Hello.
meantime में disconnect हो गया था. Now it is connected.
कनेक्ट हो रहा है. हाँ, हो गया मैम.
ठीक है तब. So
now visible.
Is visible now? Okay, thank you. So what I was talking about regarding infrastructure of state drug laboratory. So you will take an example that we have infrastructure in all the state. We have state drug laboratory. So total capacity is 31. You take an example of Jammu Kashmir or Punjab, Haryana, Delhi, Meghalaya, Tripura. So all together. So when you talk about all drug laboratory, it is 31 drug laboratories. The total drug laboratory is 31. So they can go up to capacity is 60,000 per year. Now, what about private laboratory? As I told you that there are several labs in Panchkula. So altogether, we have 300 plus private drug laboratory, where all the state, mostly in Karnataka, Kerala, or in Andhra Pradesh, or so there is a huge number of private drug laboratories are available. So they use for a commercial activity. So we have 300 plus. Now, a function of drug testing laboratory, as you say, that moment the sample is received, that there is a registration. Many of us use a software for allocation. During the survey, we use a case software, and coding is done. It is a blinded one. That moment they collect it, a sample analysis, then it was review is done, then decoding is done. Then they have to report form of as per the Drug and Cosmetic Act standard, and not whether it is standard or not of standard quality, then the legal action will be taken. So when you collect the sample by drug inspector, it's a legal sample, whether it is NSQ or there is other spurious problem. So here the standard is followed is Indian Pharmacopoeia, United States Pharmacopoeia, British Pharmacopoeia, all the Pharmacopoeia which is global acceptance we use, and it is done as per Drug and Cosmetic Act. So these are the instrument list we use in a lab, and also the capacity to do various dosage form, like you take example of tablet, capsule, ointment, or oral dehydration liquid. So eye drop, ear drop, inhalation spray. So these parameters are used. So initially when you collect and lab, it is assessed through the physical and chemical, and then through the analytical or microbiological or pharmacological. So how do you present the data? You have a descriptive one, identify is there any impurities or related substance, then you do assay. So assay is in terms of solubility or clarity, pH, or water, loss of drying. So these are the parameters, uniformity of dispersion, these are the parameters. Regarding a specific test, you go for the stability, Parisian testing, bacterial endotoxin, sterility, particulate matters, particular size, then microbial limit, and preservative efficacy. So just look at some of the data for last couple of years. So you take an example of CDL Kolkata, that number of sample, if you see they tested and how many they got. So they collected 3,164 samples. Out of that, it is 285 is found to be not of standard quality. Similarly, in 2012 and 13, they found 395 NSQ, or 2013 and 14 they found 356. So you can take a example of lab data in different CDL, like Kosoli, they found 2011 and 12 2 NSQ, then 2012 and 13 11 NSQ, and for 2013 and 14 there are 6 NSQ. So you can take an example of different CDL, how many are product has been tested, how many they could find, and you can see the trend over a period of last 10 years or 5 years. Similarly, Chennai also, it has been reported 2013 and 14, 11 NSQ. So these are the different lab, and especially in Hyderabad, they could not find any NSQ. There is nil. So this is a positive note. That similarly for RTDL also, you can take an example that in Guwahati, NSQ in 2011 and 12 is 33, then 12 and 13 is 72, and in 2013 and 14 is 87. So what about the Chandigarh? Chandigarh is 11, 6, and 27. So then you can see that quality in terms of the product, and what about the standard quality, and what about the not standard quality. So if you would see from 2011 to 12, so it maintains standard quality from 96%, 5% to 97%. So approximately 3% of not of standard quality as per the government. So what I'd like to highlight is that India has an infrastructure for assessment of the quality. We have a central support that we have 9 labs, we have 31 state, but we have 300 plus different drug testing lab. The private lab is also there. So there also it is important that all the lab who does the quality analysis, it has to be an accredited lab. Then only you should allow for quality. But anyway, when you procure the medicine, you should have a mechanism from the hospital as a physician or pharmacist also that randomly you check for the quality. So there are different aspects, because quality linked to that you have a good quality medicine, that we have a good medication management system. But at the same time, the pricing is also important. How do you make it accessible and affordable, and all the medicines are available, the good quality medicine. So that is what, though there are challenges that many of the medicines are not available in the periphery or remote places, or also you have to look at that, that how you make it, and you have to see the storage condition in order to keep the quality once it is dispersed from the manufacturing side. So that also need to be looked at the distribution phase. How do you make a good mechanism and stable one and sustainable one we do it. So we have a lot of challenges, but anyway, we have to believe that unless there is an awareness that we need to have a good quality medicine, and how do you check? Like we always say that how do you describe as a professional NSQ, not of standard quality, or you say spurious? Can you give me the example? And the counterfeit medicine, as I said, that till today counterfeit what is not included in the regulatory, but there is a issue of counterfeit also. Maybe it is a good quality or bad quality. So based on that, analysis only you can evaluate that. So in conclusion, I say that developed or in developed countries also we have challenges, but there should be mechanism to grab this problem, and so that we have a successful, the good quality medicine, and everyone should be happy that we can, do successful in a healthcare delivery system with the if we maintain the quality. Thank you very much. So I keep around 10 minutes for discussion so that we can discuss. Any questions we have? Any questions? Can you hear me?
Hello, yes, sir. Your voice is audible.
Yes, correct.
Yes, sir. Very thanks, Lord, sir. Very nice, informative session.
Can you hear me now?
Yes, sir. Your voice is audible. Thanks, Lord, sir, for very nice, informative session and providing good brief views about the pharmacy practice module. Now, I would like to invite Dr. Indrajit Verma for participant size queries.
Sir, I am audible to you? Sir, I am audible to you?
Yeah, तो sir बहुत nice information.
I think this is what the experts are for. Thank you very much, Dr. Medhi, for this informative lecture on quality of drugs. You have filled our mind with complete knowledge and information. Now we'll start our second session with Dr. Deepika Bansal in 5 minutes.
हाँ हाँ थोड़ा 5-7 मिनट कैंडिडेट्स को दे दो। मैं ठीक है मैं इतने आपको स्क्रीन शेयर करके दिखा देती हूँ। ठीक है, हाँ मैं ना ऑनलाइन ही हूँ। आप अपने जो भी आपका कोऑर्डिनेटर है ना उसको बोल दो कि मैं अपना स्क्रीन शेयर कर देती हूँ। ठीक है ओके जी।
Hello, Vikas Medhi, sir. Voice is audible.
Yeah, I can hear you.
Sir, thanks so much, sir. Very informative session. You provided brief knowledge about the pharmacy practice module. Sir, I have some query regarding from participant side. Sir, if you have
Okay,
any
but
time for 5 to 10 minutes, then we're asking about the question round.
no, what I'm saying is there any question? Then I can answer. Your audio is not clear. It is coming breaking voice.
Okay, okay, sir.
So any questions is there? I can answer it.
Okay, okay, sir. Now I would like to invite Dr. Indrajit Verma, Associate Professor in PharmD, for participant size query session.
Can you repeat the question? I'm not able to hear.
Hello, I'm audible. Audible. Okay, sir. I have a query. It was really informative information you had given. So I have a query that is there any national formulary for high alert medication in India?
Is there any national formulary for
National formulary for high alert medication.
high?
High alert.
No, we have a national essential medicine list. We do not have a separate list.
Okay, and not for even antibiotics or antimicrobial agents also.
Yeah, that has been incorporated. All the sectors are incorporated in national essential list. So it is now under revision. So now it is also included since materials are there. So it is also included. So as I say, now we call it a product. Reason behind there are a lot of materials also will be included. So there is a separate list for medical devices also.
Okay, sir. Okay. Thank you, sir.
Thank you.
Hello. Hello.
Yeah, I can because audio is not clear.
Sir, I want to ask you one question. In allopathic drugs, we have a DPSO. So I want to know there is any DPSO in Ayurvedic drugs? Yeah, in Ayurvedic formulary or even in the nutraceuticals?
Actually, the audio is not at all clear. I just heard the allopathic word and can you repeat and slowly?
हाँ एक मिनट सही है ना ऐसी है ना हेलो
Yeah.
now it is audible sir
No, that voice is coming, breaking voice.
hello now it is audible sir
I think what you can do is you can ask everybody to mute, then it will be clear. I think.
no issue sir I will ask you from I will call you sir later.
Thank you, ma'am.
No issue. Thank you sir
Hello. Thank you, sir. I think this is what the experts are for. Thank you very much, Dr. Medhi, for this informative lecture on quality of drugs. You have filled our mind with complete knowledge and information. Thank you so much, sir.
Audio is not. Can you use a chat box or so that I can get the question? Is there any system for chat box? You can write. I think there is a chat box now. You can write the question so that at least I'll get the question because my audio is clear to you. I can reply it. How many participants are there today?
Sir, more than 100 participants are there for today's session. Sir, I think no queries in the chat box.
Yeah, you can write in a chat box so that I can reply it.
So right now there are no queries in the chat box.
Okay, thank you. No problem.
Okay, thank you so much, sir.
Thank you.
We are very much pleased to have with us our next speaker, Dr. Deepika Bansal. Ma'am, are you there? Dr. Deepika Bansal.
Hello, good morning, ma'am. Hello, Deepika ma'am. Good morning.
Good morning,
Voice is
Dr.
audible?
Parvinder. हाँ जी बिल्कुल it is audible.
Okay.
How are you?
Welcome, ma'am. Fine, ma'am. Welcome.
Good. Thank you.
Okay, ma'am. You share your presentation, then I add on the screen.
अच्छा, I just share the screen or should I send you the mail?
Ma'am, maybe you have share screen, then I add on the
Okay.
screen. You have only just share screen.
Okay. Can you see?
No, ma'am. Slide is not visible.
Hello, Doctor Doctor
Hello?
Doctor
Yes.
Parvinder. Can you see? I have hello. हाँ जी,
Yes,
can
ma'am.
you see?
No, ma'am. Your slide is not visible.
अच्छा, let me try. I have actually done the share screen.
Okay.
Wait, it will come.
Okay, ma'am. Wait.
I have clicked. Is it available now? Is it?
No,
Can
ma'am.
you
It's
see now?
not. No,
Okay,
ma'am.
wait.
I not. Okay.
Can you see it now?
No, ma'am. Ma'am, if any
Okay.
difficulty, then you can mail.
Okay. Let me see. Huh? Just a second.
Okay, ma'am.
Now can you see? Hello, Dr. Parminder. Dr. Parminder.
Yes, ma'am. No. Yes, ma'am. Your presentation is not visible, ma'am. Maybe any technical issue, ma'am.
No,
You
it
firstly
is
go
not.
the share screen.
Okay. Huh. I have done that. I
Okay.
have shared the screen from my side. Actually, my window it is visible. It is
Okay.
share. I have clicked also. Mhm. If it is not now.
Yes, ma'am. Ma'am, you can mail this
Huh,
presentation.
but then how will I change the screens? Will I be able to from my own end?
Yes, ma'am. Yes, ma'am.
Okay, then just a second. Just a second. I am sending you this. I just received one mail from Dr. Inderjeet. I am replying
Yes,
to
ma'am.
that only. Okay.
Okay.
Parminder, look, Deepika Bansal and I have sent you the mail.
Yes, ma'am. Okay, ma'am. I
They say share your screen, but I think from your end something is wrong because I'm not able to click that button of share. Why is it so?
okay, ma'am. Nothing issue. I will play the slide.
Hello? 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 Dr. Parminder, can you hear me?
Voice is audible. After 5 minutes, I
हाँ जी
play
बिल्कुल
you.
audible. I think मेरी
Yes, ma'am.
मैंने अपनी स्क्रीन शेयर किया और वो शेयर भी हो रही है.
Yes, yes, ma'am. Your slide is visible.
हाँ, मैं अभी स्लाइड शो कर दूं क्या?
Wait, wait for 1 minute, ma'am, please.
We are very much pleased to have with us our next speaker, Dr. Deepika Bansal. We welcome you, ma'am. Dr. Deepika Bansal is working as an assistant professor in clinical research unit, Department of Pharmacy Practice, NIPER Mohali. She did her MD and DM clinical pharmacology from the prestigious institute PGI, Chandigarh with significant distinction. Her research work is focused on clinical trials. She is a reviewer and editorial board member of many international journals. She has authored over 100 peer-reviewed publications and she has been awarded with the national level Young Investigator Shakuntala Amitchand Award in 2015 by Indian Council of Medical Research. We welcome you, ma'am.
Thank you so much. I hope I am audible.
Yes, ma'am, your voice is audible.
Dr. Parminder, am I audible?
Yes, ma'am, you are audible. You can start the session, ma'am.
And
Your
the
slide
slides
is
are
visible.
visible?
Yes, ma'am,
Okay.
your slide is visible.
Okay, so
Okay, thanks, ma'am.
a very good morning to all of you. First of all, I would like to thank Dr. Inderjeet, Dr. Parminder for giving me such a nice introduction and inviting me to deliver this talk on the advanced pharmacy practice module for healthcare professionals. And as I just heard from Dr. Inderjeet, more than 100 participants have joined this webinar. So I welcome all the participants and hope the session is going to be very, very useful to you. So before we start actually the scientific session, let me brief the few learning objectives that you can keep it in your mind from the present lecture. The first important thing is we will try to understand what is a systematic review. And as from my previous experience, many of the students get confused between the differences between systematic reviews and traditional reviews, or what we call as a narrative review. So we will also try to understand the difference between a systematic review and a traditional review. Of course, we will try to understand the importance of systematic reviews and meta-analysis. What are the steps in performing a systematic review or a meta-analysis, and the various points that are included in creating the report of a systematic review. So before you understand how do we conduct or what is a systematic review, let us first try to understand this colorful picture. There are a lot of things in this colorful picture that we have to understand. The first important thing, if you can see the shape, this is a kind of a pyramid-based shape. This is called as an evidence-based pyramid, or also known as a wedge of pyramid. Wedge of evidence. If you see, we starting from the base going to the top, we see that the meta-analysis and the systematic reviews are on the top of this evidence-based pyramid. But that does not mean that only the systematic reviews and the meta-analysis consist of the evidence in clinical research or pharmacy practice or medical sciences. We see that even the individual case reports, even the in vitro reports, expert opinions, some kind of a background information, animal studies, cell culture studies, everything gives us a piece of evidence. This becomes all the more important since healthcare science is a vast science. And before venturing on to discovery and the development of specific therapies, we must have a lot of background information. So you see that even the individual case reports, what we call as a ICSRs, also becomes an evidence, though they have got lesser quality. Then a series of case reports becomes a case series. Then we have few observational studies in which we do not intervene. So these are called as cohort studies or case control studies. Then we come on to the interventional clinical trials. These are of two types. One, which has got a lesser evidence. These are known as non-randomized controlled clinical trial. And on the top, we have a better quality of evidence, which is known as randomized controlled clinical trial. And from these, we generate something known as critically appraised literature or evidence-based practice guidelines. And from these critically appraised literature, which is basically this primary literature consisting of in vitro reports, case series, cohort and case control studies, non-randomized and randomized clinical trials, we pool the evidence and they become systematic reviews. So all the reviews that we have consist of systematic reviews, and if some of the data is quantitative and it permits, we pool the data quantitatively using some statistical tools and we generate meta-analysis. But this is not all about this figure. We have a lot of other things in this picture or in this slide. Please try to understand. The first important thing that you need to understand is, as we go higher up from the base to the top of this pyramid, the quality of evidence or the quality of the study improves. So as we go higher up, the quality of the studies improve. This is also known as critical appraisal of the literature. Then, as I just mentioned earlier also, the two important types of studies which are considered as primary studies or the primary literature. What do we mean by primary studies? We actually interview or enroll the patients in the studies and collect the data, collate the data finally, and publish the data or generate the reports. These studies or the primary literature studies are classified into two important components. One is your observational studies, another is your experimental studies. These both observational studies and the experimental studies pooled up are known as clinical research primary literature studies. And when we already have some observational studies and the experimental studies as the primary literature evidence, and then we try to pool up these primary available studies in the literature, we generate something known as secondary literature. This is known as systematic reviews and meta-analysis. What exactly it is, we will see in the coming up slides. So we have understood something known as wedge. We have understood observational studies, we have understood experimental studies, we have understood something known as primary literature and secondary literature. Then there are two arrows that you can see. One is the LOE, which is known as level of evidence, and another is bias. Now, as you see, that as I just mentioned, that the quality of the evidence improves as we go from the base of this pyramid to the top of the pyramid, and that's how what we say that the level of evidence increases as we go from the base to the top of this pyramid. And also the bias involved in conducting the studies decreases. Bias is least when you are at the top of this pyramid, and the bias is highest when you are at the base of this pyramid. However, one needs to understand very basic point that each kind of a research study is a piece of evidence, and you cannot negate any piece of evidence while generating the final top of the wedge of evidence. So now let us try to understand what is a systematic review and what is a meta-analysis. Systematic review, so if you are conducting a particular review on a particular question, well-articulated question, and you involve systematic research, systematic search strategy, inclusion, exclusion, or the eligibility criteria, so this becomes a systematic review. And not all the systematic reviews will have a meta-analysis component. Actually, a lot of students ask me, ma'am, why do these things come together? So you need to understand that all the systematically created secondary literature becomes systematic review, but only if the data in the original studies, that is the primary literature studies, the observational and the experimental randomized or non-randomized clinical trial permits. The data is quantitative, the endpoints are uniform in 2 or more publications, only then we will be able to conduct the meta-analysis. So meta-analysis is actually an optional part of a systematic review. So you need to also understand one important point, that all the meta-analysis have got a systematic review component, but not all the systematic reviews do have a meta-analysis component. So let us further discuss the three important things in creating the secondary generation of the literature. First is the review, also known as traditional review or narrative review. Second is your systematic reviews, and third is your meta-analysis. So what is a narrative review? Narrative review, let's say as a supervisor, I give a question or some research query to a student, and I ask the student to read some literature about that research query. This research query will, hello, Dr. Parminder, am I audible? Okay, this research query is being read by the student. The student can collect 2-3 articles, 2-3 papers, 2-3 publications on that, and he or she will collect the general information, and he will attempt to synthesize few commentaries or the results and the conclusions from these 2 or more publications on this given topic. So he brings about some information on that topic, and this is known as narrative review. This narrative review, since it does not include a systematic search strategy and an eligibility criteria to include the primary literature, so this may not be reproducible. So this is known as traditional or a narrative review. Going on to the systematic review, very important definition of systematic review is that when a review strives to comprehensively identify and track down all the literature on a given topic. So this clearly shows that the systematic review will have a well-formed, articulated question which is going to be answered. So you will have a very standard methodology in which the search strategy, the conduction of the review, the inclusion of few studies and the exclusion of the studies will be documented. Shall be documented actually a priori in a well-written protocol. So the systematic review has got a very well-formed question and the search strategy, and also very step-up procedure or the steps to get it formalized into a complete report. It has to include all the articles published on that topic, while the narrative review may or may not include all the articles or the research studies that have been conducted to answer that particular question. So this is the basic difference between the systematic review and a narrative review. Coming on to the meta-analysis. Meta-analysis is actually a small component of a systematic review. What happens is that if few research studies gives you some quantitative data, that means there are specific endpoints in it which are quantitative. For example, let's say we are doing a meta-analysis of some specific anti-epileptic drug. So let's say we have there is a drug known as topiramate, and it is going to be used in generalized tonic-clonic seizures. So for GTSS seizures in adults, the meta-analysis should have a specific quantitative outcome, let's say 100% reduction in the seizure frequency in the patients. So this endpoint is going to be pooled using a very specific statistical strategy, giving us a meta-analysis concept in the systematic review. To further understand, please see this picture and see that meta-analysis is a very small component. While when you search in the huge databases like Medline, PubMed, all the reviews that you get are narrative reviews. These are all the reviews. And among all these reviews, only a very small component, the yellow component, which is a systematic review. And among these huge number of systematic reviews, only a very, very small component would have pooled the quantitative data, giving us the concept of meta-analysis. So please try to understand that meta-analysis may be a part of few systematic reviews, but all the meta-analysis may not be a part of the systematic reviews, while all the systematic reviews are the ones which have got a specific search strategy and should have included all the papers published on that particular topic. So actually, this is a slide which I just have included, and this slide gives you a lot of differences between a narrative review and a systematic review. Why I am trying to give you a lot of importance on that systematic review. First, please try to understand that systematic review, though considered as a review, but actually it is in itself a huge research. Sometimes it's that bigger research that it may even take 2 to 3 years to complete one systematic review. Now, if you go to the highest the most stringent types of systematic reviews, that is Cochrane systematic reviews, you will see that it is always 3 to 4 members that are involved in one systematic review. The members can be more, but there cannot be less than 2. And the second important thing, it takes a minimum of 2 to 3 years to get one Cochrane systematic review to get published. So let us try to understand in detail the differences between narrative review and systematic review. The first important thing, you need to see whether the methodology section is there. What does the methodology section consist? The methodology consists of clearly described protocol with detailed method. What does it mean? It means that well-framed research question. This research question should be replicated in your search strategy. The search strategy, as we will see in the coming up slides, consists of mesh items, keywords, clearly defined question, how you have formulated that search strategy, is it reproducible? So that means once you have created one search strategy, it could be reproduced again and again, done with using the same search strategy by various researchers. Second, the articles included. So after performing the search strategy in various databases like Medline, Embase, etc., you should get the same number of articles, the same type of articles, the type of articles that the primary authors have. You should have written clearly the methodology, including the inclusion criteria, what kind of studies you are going to include, what kind of studies you are going to exclude, what are the statistical models that you are going to use in creating the systematic review or pooling up the results of the primary studies, while no such methods are being described in the narrative reviews. Second important difference between the narrative review is that in the narrative review, you may include, you may not include all the articles published in that particular area. So you may just search one database, that is usually Medline, which is freely available to all the researchers. So this usually leads to the publication bias. Again, we will try to understand the publication bias in the coming up slides. While in case of systematic review, you have to perform a comprehensive literature search. So it is usually the guideline suggests that one database is not sufficient to perform the systematic review. So minimum 2 to 3 medical databases one needs to search to collect all the studies published in that particular research question. What is the third difference? The third difference is you do not give credit to the study design. Study designs means you can actually pool the observational studies as well as the experimental studies, that is cohort and case control studies can be pooled with the controlled clinical trials in case of a narrative review, which gives just the commentaries, while the systematic review is very particular and very stringent that you can perform the systematic review of either randomized control trials or you can perform the systematic review of observational studies. Not only that, you also try to evaluate the validity of each individual clinical study which is being included in the systematic review. The systematic review has to be a quality systematic review. For that, the included studies should be of good quality or should have very high level of evidence. So one needs to perform the qualitative as well as the quantitative methods of validity assessment of individual studies. We will see that how do we assess the validity of each individual study by using the method known as critical appraisal. And this critical appraisal involves what kind of tools that we will see in the coming up slides. So the next important thing is that it relies on the p-values. P-values of what? P-values of individual studies that are included in the narrative review. While if you see the systematic review, it takes the effect sizes or the actual data from the primary studies and it creates its own p-value by pooling up the individual sizes along with the confidence interval. How do we read the p-values along with the confidence interval? We will just see. So the next difference between the narrative versus the systematic review is that the narrative reviews overemphasizes on the vote counting. Vote counting, each trial is given unequal weight. So that means if we are included 5 different studies in the narrative review, all the studies are being given equal amount of weight. What is the actual meaning of weightage in the statistical terminology? I'll explain you. Well, if you see the systematic review and I'll show you some forest plot, you will try to understand, you will see that all the studies included in the systematic review are not given equal weightage, while some studies are being given more weightage and some studies are being given less weightage. We will try to see what is the exact meaning of weightage. So narrative review is basically more of a descriptive in nature, while systematic review involves a component known as meta-analysis, as we just understood in the previous slides, which uses some kind of statistical strategies and tools, and it pools the results of similarly conducted original or the primary clinical studies, and it creates a tower of power. Finally, we will see that this is the pictorial representation of traditional versus systematic review. In case of traditional review, you can pool apples and oranges, while this is not so in case of systematic review. In the systematic review, you either have the pooled up results of only the oranges or the pooled up results of only of green apples. And this is the Cochrane determination of the systematic review. This is a pictorial representation, which you see that it is a kind of a funnel approach where initially from the search of the studies from various databases, you can see that there are various kinds of studies. Here in this picture, it is being represented by some triangles, oval-shaped structures, square-shaped structures, blue, light blue, oranges. So initially, from when you perform a search in a medical database, you will get lots of types of studies which are being conducted using various study designs as being observational, as being experimental. Observational also you can have prospective, retrospective, while in the experimental you can have non-randomized studies, randomized studies. Some are the studies which are having very good quality of evidence, some are the studies which are biased studies. So you do not pool all the studies together, which are usually being done in the traditional review. You try to extract the data, you try to sift, you try to use a kind of a sieve, and you sift and select the studies according to your eligibility criteria, and you pick up only the studies which are conducted in a uniform manner or similar manner, and then you try to extract the data from these studies, and finally you get a very uniform systematic review which you try to create, collate and synthesize the information and publish. Okay, So now let us try to understand the important component of the systematic review, that is meta-analysis. Meta-analysis is the most important quantitative component of the systematic review, because actually it uses a lot of statistical tools and statistical strategies which gives you a very high powered answer to your well articulated, very narrow research question. The four important steps that are being used are: one, the identification of studies, as we just saw in the previous slides, which kind of studies you are going to include, what is going to be your search strategy, what is going to be your search engines to find your studies, and then the second thing is you determine the eligibility of the studies. Even if you decide that you are going to use only the randomized controlled clinical trials, but are you going to use all the randomized controlled clinical trials published on that particular topic? No. You always create a very stringent inclusion criteria to guide you that these are the studies which we are going to keep in the systematic review, and these are written or well documented in the protocol of the systematic reviews. Then you also have some exclusion criteria, that these are the, if the study has got this component, we are going to discard these studies and not include in our systematic review. After freezing that these are the type and the number of the research studies we are going to include in our systematic review, you abstract or get the data from the original studies, and you create your case record forms, which is usually an electronic format in the MS Excel. You also try to evaluate the validity, which is known as critical appraisal, using various scales or tools, and this is the way you abstract the data from each and every study to be included in the systematic review. It's not that you are going to include all the information. You create very specific data items, and these data items are being that you are going to be included in the systematic review. Finally, you use the statistical tools to analyze the quantitative data which is present in each individual study. What is this quantitative data? This is regarding, like, you can, if you are evaluating the efficacy, so it can be like efficacy of a drug, safety of a drug. Usually the endpoints that are being analyzed in a meta-analysis component, or being pooled in a meta-analysis component to get your pooled effect estimate, or to get the outcome in the systematic review in a quantitative manner, is usually you can have efficacy outcome, you can have safety outcome. Nowadays we are also having a lot of outcomes known as health related quality of life outcomes also to be pooled in a meta analysis manner. So the first important component, identify the studies. How do we identify the studies that are going to be included in your systematic review? The crux is that you need to be methodological. So that means you need to plan ahead. It is not that first you get the studies and then you plan. No, like just the way you write your protocol for clinical trials, for cohort studies, for case control studies, the same way you write a protocol for conducting a systematic review. So the first important thing, you need to create a question, and then you create your search strategy, and then you put that search strategy in the various medical databases to perform the search. So let us first see what are the various databases that we use to perform the search to identify the clinical studies or the original clinical studies to be included in the systematic review. The most important database that everybody is aware about is the PubMed. Medline is the major component of the PubMed. This is freely available. You know of this website. Everybody knows of this website. You can have the two important things in this is that in the PubMed we have a PubMed Central. PubMed Central gives you the full text article, while not all the articles articles are available in a full text manner. That means full PDF, full study is not available usually on the PubMed, but PubMed is still a very popular and very vast medical database because you get the maximum number of study titles along with their publication details, and majority of the articles do give their abstract of around 200 or 250 words about the study, which gives you the actual nature of the study in a gist manner. So the PubMed is must to be included in the search strategy before you plan any systematic review or on a therapeutic strategy or a diagnostic strategy or a preventive strategy. But is that sufficient? No, it is not sufficient. PubMed cannot be just included in the systematic review. It can be just one of the medical database that you can include in your systematic review. You are supposed to include again a minimum of 2 to 3 medical databases to identify, to search for the appropriate studies to be included in your systematic review, so that you do not actually miss any study which has been conducted in that particular question or a particular topic for which you are going to perform the systematic review. So another database which is very popular nowadays is the MBase, and this is available through these website. MBase is a database which involves a little bit different search strategy, but the important limitation for using MBase is that this is a paid medical database. You need to pay even to perform the search in this kind of a search engine. It is available in major institutes across the world, so you can actually take the help of few institutes, or you can collaborate with those institutes where the MBase subscription is there if your institute is not having the subscription of MBase. But MBase is one such database which gives a lot of original articles which are actually not there in the PubMed. The third important thing is the Cochrane library. Now Cochrane library or the Cochrane database is a very a favorite database of the people who are actually involved in randomized controlled clinical trials. Cochrane has got two components, one for the systematic reviews and one for the clinical trials, because the Cochrane Colloquium performs systematic reviews on their own also. So so you just have to go to the Cochrane website, then you have to click on the Cochrane Central, and from the Cochrane Central you can get a lot of clinical trials, RCTs, randomized clinical trials, which are being performed in the topic which you are interested to perform the systematic reviews. And one good part about Cochrane library is that if you are residing in India, then this library is free to you, because we have the subscription throughout the country for the Cochrane library. So Cochrane library is a must to be included. It is freely available to all the researchers in India. You can go to the Cochrane Central to get the clinical trials, or you can see the systematic reviews to actually see if already some systematic review on the topic of your own interest has already been conducted. So this is about the Cochrane, and we'll see something more about the Cochrane in the coming. So do you think this is sufficient? No, we have a lot of other databases like Ovid, like ProQuest, Elsevier, Scopus, EBSCO database, Hinari databases, these database, Sinahal database. So there are a lot of databases that you need to go depending upon your research question. And then let's come on to the other strategies. The first important thing is the hand strategy. Do you think that hand search is important now? Well, nowadays, yes, it is important. There are a lot of journals which are not PubMed indexed. There are a lot of journals which are not even MBase indexed. So you will not get all the papers in these two databases, and many a times you have the subscription in your library about some national journals or some South Asian journals which are giving you the articles on the research topic which is particularly of a South Asian region. So as far as our students are concerned and performing the systematic reviews of the research questions which are of particular interest to the South Asian region or the Indian origin, I will definitely suggest that please go for the hand search of all the journals in your area of interest from your library to see whether some papers of the related to your research question are being published. Another important thing that you need to actually perform in each systematic review is you need to see the cross references. That means from the initial search from PubMed, Embase, Cochrane, you would get, let's say, 10 articles. Please see the bibliography or the reference articles of all these retrieved articles. So checking up the references of all the retrieved article, which is known as cross citations of checking the bibliography of the retrieved articles, you are bound to get lot of new references, new articles that can be included in your systematic review. All these things to perform this extensive search is important, because systematic review cannot exclude any particular research article if that is of interest to answer that particular question. Of course, you can look for the Google Scholar, which is a very large database, and of course a vast database. A lot of freely available full text articles are also available through Google Scholar. It is available to almost all the researchers, and one has got not only the access while the surfing expertise for the Google Scholar. Now, the last thing is the unpublished literature. So that means while these are in the conduction stage or in the final reporting stage, but yet these have not come in your peer-reviewed journal. The first important question: do we need to include this? The answer is yes as well as no. The answer is no if the question is well framed, it is available in the literature since many decades, you have enough of published literature, so you may not include the unpublished literature. But the answer is yes, you need to include the unpublished literature if the research question you are going to answer in the systematic review is a hot burning topic. The example being COVID-19. You know that the COVID-19 is at present very hot burning topic, very topic of interest to almost all the researchers. A lot of research across the globe, worldwide, it is being performed, and not all the research is being published. A lot of the research is in the conduction stage. So one might need to include the unpublished literature in such kind of questions. Where do we get the unpublished literature? Now the important question arises. The first is ICTRP, International Clinical Trial Registry Platform, which is being maintained by WHO, which registers a lot of clinical trial across the globe. If you are interested in the clinical trials which are being done in US, you have clinicaltrials.gov. And the third is CTRI, Clinical Trial Registry of India, which gives you Indian specific clinical trials. Now, after including the unpublished literature, all the researchers, students who are interested in including these kind of evidence in your systematic review must always keep this thing in the mind that unpublished literature may not be of a good quality in nature. Why? Because these are not peer reviewed. These have not undergone through stringent, deep, extensive appraisal by the reviewers and the editorial board members. And before publishing, you know that you have to undergo a lot of processes, editing, peer reviewed. So unpublished literature have not undergone through these steps. So the quality may be compromised in case of unpublished literature. So you must be very cautious if you are planning to include the unpublished literature. This is also true for including the abstracts from some conferences. There are a lot of conferences which publishes the results in the form of the published posters and the oral presentations which have been done in their conferences in the journal special issue. But again, if you are including these kind of abstracts in your systematic review, you need to be very cautious, because these abstracts again may miss significant pertinent information. Second, they have not undergone through the stringent peer review process. So again, those kind of studies, that is the conference abstracts, may be published, but they lack the specific peer review process, so the quality is definitely compromised. Now, this is an example. I want to make you understand that how do we perform the search. Earlier slide we just saw what are the databases. So now you need to perform the search in the database. How do you formulate the question? That is the most important thing. The question is formed in the form of PICO, or which is known as PICO. Nowadays we also include S in the last. This is known as study design. So let us try to understand this question. In a 65-year-old male patient with type 2 diabetes mellitus and established cardiovascular disease, will empagliflozin, which is an SGLT2 inhibitor, compared to placebo, prevent the death from the cardiovascular causes? So you can see that here the patients with the type 2 diabetes and established cardiovascular disease becomes the P, patients, also known as population. Empagliflozin becomes your intervention. Compared to what? Placebo. So placebo or some other drug may act as your comparator, or a C in case of PICO. And what is your outcome, endpoint that I was talking about in the other slide, to create your meta-analytic component in the systematic review? Prevent the death from the cardiovascular causes. So it is like mortality from cardiovascular causes becomes your outcome. So these are the 4 important things that you are going to frame in any kind of search strategy. So these search strategies are going to be included as keywords in the column for putting up the search strategy in any of the major search engine medical database. But here the important thing that you need to understand is you can't just put the whole of these 3-4 sentences in the search strategy box, because this is not going to fetch you or give you any kind of research, original research papers that you can include in your systematic review. You need to create a well-formed research search strategy that you can include, that you can put in the search engine and get the desired articles. So how do you put? You can put like diabetes and cardiovascular disease and empagliflozin and death. But does this end your search strategy? No, the search strategy doesn't end here, because you need to include all the synonyms of all these 4 points, that is PICO. Type 2 diabetes, some of the studies may mention only diabetes, some of the studies may mention only diabetes mellitus. So all these 3 things, type 2 diabetes, DM, diabetes mellitus, may act as synonyms to enter into your search strategy. Second, cardiovascular disease. You can have CVS disorders. You can have heart diseases. So this can become as your another synonyms for cardiovascular disease. Third is your intervention, that is empagliflozin. Somebody may include the term SGLT2 inhibitor. Somebody may include some trade names for this drugs or the brand names for this drug. So all these drugs you have to find, all these keywords you have to find. This is also known as MESH or medical subject heading, and you need to put in your search strategy. Finally, you can put placebo, or you may exclude this placebo. Outcome also you can put morbidity, mortality. So that you do not exclude in your initial search strategy and you are able to find the maximum number of articles while performing the search strategy. I'll just show you how the search strategy is being written. If you can see this slide, this is from one of my research papers where I was trying to perform a systematic review in case of diabetic ketoacidosis. So here you can see the word is written DKA. And we use three kind of Boolean operators in case of the performing search strategy. This is a search strategy performed in MBase. This needs to be mentioned, and this needs to be very clear in the mind that the methodology of conducting the search strategy, writing of the search strategy is different in all the search engines. So the search strategy which is being performed in PubMed is a little bit different from MBase and Ovid. So if you see that initially we performed this step 1 and we got 17,617 articles, and then we put some kind of limitations, and this intervention is being added in the second step, and this leads to this many number of articles, 759019, and then we wanted just to include the S, that is clinical trials only, and so we have added the terminology known as randomization, placebo, double blind, controlled clinical trials, and we have got the number. See, the number is increasing from 17,000, so now it is crossing 1 lakh. And finally, we wanted to see the efficacy, toxicity, safety of various insulin analogs that you can see, Lantus, Glargine, etc., etc. And even we finally combined the 4 search strategies, that is PICOS. We got 874 titles. Now this is titles, and from here the search strategy is being frozen, and these 874 titles will now be read thoroughly to and apply, and you will apply your inclusion and exclusion criteria. That is your second step that we will see in the coming up slide. So now the second step is the eligibility criteria. Now here you need to keep in the mind that you cannot include all the 874 articles because these may not fit into your inclusion and exclusion criteria. You include some articles, you exclude some articles. How do you include? Based upon your PICO. The way you have created your PICO, you freeze your inclusion criteria. So that means you here, let's say in the DKA kind of a study, I just wanted to include only the randomized controlled clinical trial, right? I do not wanted to include observational clinical trials. Like in the previous slide, I wanted to include only empagliflozin and not other SGLT2 inhibitors. Now this might be used, this SGLT2 inhibitor might be used as an hypoglycemic agent, but I wanted to see the cardiovascular disease related outcome in patients who are suffering with type 2 diabetes mellitus. So these you can keep in your inclusion criteria. And similarly, you will keep some kind of an exclusion criteria based on which you are going to be excluding those studies. The very important criteria that we keep in the exclusion criteria is one based on the study design. So if I am interested only in including the randomized controlled clinical trial, certainly I want to exclude all sorts of case reports, case series, some kinds of observational studies like case control or cohort studies, some editorials, some letter to the editors, some kind of primary reviews, narrative reviews. Though narrative reviews can act as a very good source to retrieve the primary original studies, that is RCT, but as such the data from the reviews are not going to be included in the systematic reviews. Here I want to make another point clear to you, that here I wanted to include only the randomized controlled trials, but this may not be true in all sorts of random systematic reviews. In some of the systematic reviews, you want to pull observational studies. So for that case, the the component will not be RCTs. The component will be observational studies, including case series, cross-sectional studies, case control studies, cohort studies. So how will you decide you want to include observational studies or randomized controlled clinical trials? This entirely depends upon the type of question that you are interested, the type of question that you are going to answer in your systematic review. So the important thing is that you need to frame the study eligibility criteria, which includes the inclusion criteria, which also includes some of the exclusion criteria. The inclusion criteria and the exclusion criteria needs to be mutually exclusive. You cannot have some kind of superimpositions. A very simple example here I would like to give. For example, if you choose that you are going to choose adults in your primary studies, so you cannot have here also adults. So here you will have adults. So in the exclusion criteria, you will have children. Or you also can have elderly, depending upon how stringent, how narrow your systematic review inclusion and exclusion criteria is going to. Okay, so now let's come on to the important checklist that is known as PRISMA checklist or PRISMA flow chart. If some of you are aware about the consort statement that is being used in randomized controlled clinical trial, those of you can very well appreciate what is PRISMA flow chart. PRISMA flow chart is nothing but it is the algorithm in which you show how you have conducted the various steps in performing the systematic review. So this is basically a flow of information through the different phases of the conduction of the systematic review. So again, the same important 4 components: identification, screening, eligibility, and finally you have included what kind of studies in your systematic review. So in the identification process, you have various databases that you are going to report, and from each database how many studies you have got by applying the search strategy. Here an important component I would like to mention, that nowadays the journals or the reports also wants your search strategy to be frozen and submitted along with your systematic review report also. So be very much clear and document everything right from the first step of creation of PICO and the search strategy. Let's say you have identified 2 important databases, PubMed and Embase, from which you are going to perform the search strategy. So let's say here you have found from the PubMed 100 records, and here from the Embase you have found another 100 records. I'm sure that while you will perform the search strategy using the 2 databases, there will be a lot of articles which you will find in both the search engine. So here you will get some of the articles which are present in both the papers. This means these are duplicates and you cannot include them in your systematic review in a duplication manner. So the important component here is that you need to remove the duplicates from which you have got from both the search engines, and you are going to include only once the papers which you have got in both the databases. From these records which you have got as a titles, you will read the abstracts, and you will see that some of the records still, after reading the abstract, you will see that though the titles match your PICO, but in the abstract you do not have the actual information that is being desired to be included in the systematic review. So here you need to write that how many articles you have excluded after reading the abstract of the various titles which you have got in the initial search. After which you will have certain number which you after reading the abstracts you will have certain number. This you will fetch the full text articles, and these full text articles you will go to the methodology, because these are the original articles. They would have clearly mentioned the inclusion and exclusion criteria in their methodology section. Read out the methodology, clearly identify the methodology in the original papers, apply the eligibility criteria of your systematic reviews, and see which are the papers that are eligible to be included in your systematic reviews and which are the papers which you need to be excluded. Here an important point is written is that with the reasons. So you need to write that these are the number of articles that have been excluded after reading the full text, and you have to provide the reason. The reason can be like it was observational while we wanted to include clinical trial. It was a narrative review. It did not provide the endpoint which was desired. It provided median values instead of mean values, something like that. But you have to define the reasons. And finally, you will write the number of studies which are pooled in the systematic review, which is usually in a kind of a qualitative synthesis. And from this you will see how many studies provided you with the quantitative data, the endpoint which is desirable, which is specific in nature for a particular intervention, and these are the studies which are being collated to synthesize the quantitative evidence, or these are the number of the studies, or these are the number of the studies from the systematic review on which you are going to perform and report the meta-analysis results. So this is the PRISMA flow chart. You need to mention, you need to report clear each and every step of PRISMA flow chart while reporting the systematic review. So the initially I just mentioned you have to report the search strategies that you have used in various search engines, and then you have to report the PRISMA flow chart, how you have how the information flows from first step to the last step in the systematic review. Now, the third important step is abstracting the data. Now, here, the ones who have performed the original studies like observational or clinical trials, everybody knows that we create something known as case record form, on which we document the data which we get from each and every patient which is recruited in the original study, whether it is the study is observational in nature or it is a clinical trial in nature. Similarly, we have a case record form in case of systematic review, in which instead of each patient, each study becomes the each patient, and we document document the data from each study in that case record form. Now, this case record form is usually electronic in nature, and we use MS Excel to create the case record form. This is also known as spreadsheet. I think everybody is aware about that. We have columns and rows. In the columns, in each column, one item is being included. Now, what are the items usually that are being extracted from each study? Now, here you need to understand that we abstract the data initially from reading the titles and the abstracts, and then for the eligible studies that are finally going to be included in your systematic review and the meta-analysis, the data is going to be abstracted from the full text papers. Now, what are the types of data items that you are going to abstracting from each study? You are going to understand what is the journal that has published that study, which is the year of publication, which is the year of conduction of that study, the authors, the study design. Study design, you have to be very, very elaborate. For example, in case of randomized clinical trial, you need to mention randomization and the detailing about the randomization, like who conducted, was it simple randomization, block randomization, stratified randomization. You need to define, write or get the data from the original study, that how many patients were initially randomized, then was it a placebo control, was it an active comparator control, what was the intervention, what was the doses of interventions, what was the outcomes, how many patients were finally analyzed, then what is the type of analysis that you have. Type of analysis means you can have intention to treat analysis, you can have per protocol analysis. You are not going to say that this is you are not going to assume anything. If it is mentioned in the original paper, only then you are going to document that in your spreadsheet or the Excel sheet. And then, of course, you are going to also include the details about the drugs, comparisons, then the dosages, schedules, events. Now, events, you can have efficacy events. Now, it is not necessary that you just have one single efficacy endpoint. You can have multiple efficacy endpoints from each paper. You can have 3, 4, 5 endpoints. So you need to understand what is the primary efficacy outcome, what are the secondary efficacy outcomes, what are the tertiary efficacy outcomes. But you need to to get, you need to extract all the efficacy outcomes which are documented in the original research paper. Similarly, you can have the safety outcomes, and you need to put different columns for the outcomes in case of intervention arm and also the outcomes in the control arm. Now this is very important because in the coming up slides I will show you that how do we compare the intervention arm from the control arm and get the pooled effect estimates. Okay, so these are the guidelines I was talking about. This is the logo of Prisma flowchart, and this is the consort statement that you document for the clinical trials. Very important to understand here in the clinical research is what is not documented is not done. So it is essential that while performing your systematic review, you document each and every step clearly, and nothing is being reported from your own side. Whatever you have done, you have clearly reported. The way you report the things in the clinical trial, and what is the aim? This is basically, the aim is to bring the transparency while reporting any kind of a study. And this was what I was trying to tell, that systematic review in itself is a huge research, though we call this as a reviews. Now let's come on to the next thing. So we this is to evaluate the validity of the studies. So you have different kinds of tools. These are qualitative in nature. So first important tool we saw in the previous slide, that is consort, consolidated standards of reporting trials. This is basically for the RCTs. But as I said, that you can perform systematic reviews for any kind of studies. For the observational studies, that is cohort studies, case control studies, cross-sectional studies, you have something known as STROBE, that is strengthening the reporting of observational studies in epidemiology. This is similar kind of a flowchart as I showed you in Prisma, and will guide you how to report your observational studies. Similarly, you have for diagnostics tests, that is STARD, standards for reporting of diagnostic accuracy. You have some kind of a qualitative studies typically which are used in psychology studies, or nowadays for a lot of health related quality of life scales also being used, qualitative studies. For that you have something known as CORIO, consolidated criteria for reporting the qualitative research. Prisma, we have already understood, preferred reporting items for systematic reviews and meta-analysis. And if you are typically just doing a meta-analysis, you have MOOS also, which you can also include in the meta-analysis of the observational studies in epidemiology. And this is another scale that is quantitative in nature, where you can actually perform the quality assessment of the observational studies that you are going to include in your systematic reviews. This is known as Newcastle-Ottawa scale. This is freely available. This you have to use if you are doing a systematic review of lot of observational studies. So to evaluate the quality of all the included observational studies, you need to assess the quality, and this you can assess through using Newcastle-Ottawa scale. This is a quantitative scale, while the above things are known as the qualitative scales, or the most importantly they are known as checklists to report the various kinds of studies. And you can get from all these scales freely available from equator.network.org. This is the website. You just click and you will get all these checklists, and this will guide you to report the various types of studies. And these are the specific tools that are used for the quantitative assessment of the quality or the risk of bias, as we saw in the evidence-based pyramid, that as the quality of evidence improves, the risk of bias decreases. So these are like inversely correlated. The risk of bias increases, quality decreases. The quality increases, risk of bias decreases. So these are available from the logo. You can see that it is available from the Cochrane. This you just click and you will get the scale. These are quantitative scales. And there are 3 scales that are available. One is the Roby scales. This is risk of bias in the systematic review. Let's say you get a systematic review already published and you want to understand, you want to evaluate, you want to analyze that this systematic review is of quality or this systematic review is badly conducted. You apply this tool and you get the quantitative results that whether this systematic review is transparent or is it full of bias. Then the second tool is Robbins 1 tool. This has been recently introduced by Cochrane. This is to assess the risk of bias in the non-randomized studies of interventions. And finally, a lot of you might be aware about ROB 2.0 tool, which is the revised tool for risk of bias assessment in the randomized controlled clinical trial, which is considered as the highest evidence. And if you want to perform the systematic review of RCTs, you are bound to use the ROP 2.0 tool for the quality assessment of the individual studies. And this is the way it looks like, ROP 2.0 tool. And you see that the important points that are being included is one, random sequence generation to understand the selection bias. Second, the allocation concealment, again to understand or to evaluate the selection bias. The next two components are to assess the blinding of the participants in the personals, and the second is the blinding of the outcomes. So you understand that allocation concealment is also an important component for the transparency, and the blinding of the outcomes, which is very important to minimize the detection bias. Then how many people were entered in the clinical trial and how many people for which the outcome is being documented. So if the outcome data is incomplete, that will tell you the attrition bias. And finally, you will see that what kind of analysis. Is it intention to treat analysis? That is, all the patients that have been randomized are being used to assess the outcomes. This is a kind of reporting bias, and you can have some other sources. And here you see that this red says high level of bias, and you have other two which may be like not reported, or this is minimum bias. So you have three colors which says that different levels of bias in the individual studies. You can have this kind of a picture also. These both the graphs you get automatically if you use Review Manager, that is the meta-analysis or the systematic review software, which is available through Cochrane. And this you can see actually that the risk is being divided based very much quantitatively. The risk is considered as low if it is the risk of bias is low if it is green in color. The risk of bias is very high if it is red in color, and if it is light green in color, then the risk of bias is of intermediate nature. And this again tells you from the same picture as shown in the previous slide, the risk of bias from each available study you can have by just putting the data. You will get automatically this kind of a graph from the Review Manager RevMan software, which is available freely for conducting any kind of meta-analysis and systematic review through Cochrane. Okay, so now before we enter into the forest plot, please try to understand that we have already got three important components: identification of the studies, selection of the studies, and checklist quality assessment. So finally, we will perform the quantitative data analysis, or we will try to analyze the data statistically. So these are the measures of outcomes that you usually see in the individual studies and in the pooled up meta-analysis or the systematic reviews. There's an important term which is known as effect size. Effect size is nothing but this is the outcomes which we get from one group minus or divided by the outcomes from the available in the control group. For example, in the original paragraph for the identification of the studies, I showed you that empagliflozin used to decrease the cardiovascular related mortality in patients with type 2 diabetes. So the outcomes that we get in the empagliflozin treated patients minus the outcomes that we get in the control group will become the effect size. So we get the effect size from each study, and we pool these effect sizes available from each study, and finally we get the pooled effect size in systematic review or meta analysis. These effect sizes can be available in the form of relative risks, odds ratio, or hazard ratio. But is this sufficient? No, this is not sufficient. You need to have 95% confidence interval along with your point estimate, which is known as point effect size, and along with that you have 3-4 more tests that needs to be performed in case of meta-analysis, that is tests for heterogeneity, estimating the summary effects, interaction tests, and publication bias. The kinds of graphs that you get in a meta-analysis are two forest plots and funnel plots. Let us see those two. So before that, just try to understand this is known as 2x2 table. And before I show you the forest plot or I show you the effect estimates, please pay attention to this. You have two important things. One is your outcome and another is your disease. So you can have a disease, or here you can also have an intervention, disease oblique intervention oblique risk factor, right? So if you have empagliflozin present, empagliflozin absent, so you divide the patients, always take comparative two groups. There can be some patients who are exposed to the intervention, some patients who are not exposed to the intervention. Then finally the outcome, cardiovascular related mortality, which can be present, which can be absent. So here A is the one in which the intervention was there and also mortality was there. While intervention was there, the mortality was not there. In case of C, the here you need to understand that this is a control group. The intervention was not present, but still some patients did not observe cardiovascular related mortality, while some patients have cardiovascular mortality. The two types of studies that we conduct usually is one case control studies or cohort studies. Here you please understand that cohort studies go hand in hand with clinical trials, because though the cohort studies and the clinical trials both are prospective studies, so that they go forward in nature, ahead in nature. So you give the intervention today and you assess the outcome maybe 5-10 years later, maybe 1 year later, maybe 2 years later, depending upon the timelines of your study. While in the case control study, as you can see, you start from the outcomes. That is, you start from the cardiovascular related outcomes, and then you go in the past medical records of the patients, those cases who died from the cardiovascular related mortality and those cases who did not die from the cardiovascular related mortality. This becomes all the more clear in the next slide. See this. So let's say we have some patients who are having hypertension, and we know that the cardiovascular disease is an important outcome for the long-standing cases of hypertension. So to make you understand what is relative, to make you understand how do we calculate the relative risk and odds ratio. So here let us try from the left-hand corner of this 2x2 table. ABCD is known as 2x2 table. So we divide the patients into two important groups, the one who are having hypertension, that is hypertension is present, and the one which are not having the hypertension, that is disease present and the disease absent. So, and we follow these two cases for let's say 10 years or for a decade in coming in forward direction. So here we took hypertension cases in 2020, hypertension and absent hypertension cases in 2020, and we observed the outcomes in 2030. What is the outcome? Presence of cardiovascular, heart-related disease or cardiovascular disease, which can include peripheral vascular disease, CHF, myocardial infarction, etc. So we see that we took almost 493 patients having hypertension today, and we took 1271 patients who are not having hypertension in the present. And after 10 years, we saw that 90 patients who were having, among 493 cases who were having hypertension in 2020 suffered from the outcome in 2030, while 403 patients did not observe this kind of an outcome. In case of control group, that is the patients who are not having the hypertension, that is 1271, among them 70 patients observed the cardiovascular related mortality and morbidity, while 1604 cases did not observe any kind of cardiovascular mortality. So we calculate the effect sizes of effect estimates in two important terminologies. One is your relative risk, another is your odds ratio. Now, briefly, I will just mention here that relative risk is being calculated in the prospectively conducted studies, that is cohort studies or RCTs, while in the odds ratio is being calculated in cases of retrospective studies or case control studies. The formulas are very easy. The relative risk is being calculated as A upon A plus B divided by C upon C plus D. So here the relative risk for this 2 by 2 table comes out to be 3.3. While in case of odds ratio, it is calculated by A multiplied by D divided by B multiplied by C. So if you calculate the odds ratio here, it comes out to be 3.8. So let's say if this would have been conducted in a case control manner, you would have calculated odds ratio and the odds ratio would have come to be 3.8. But do you think that this calculation is sufficient? No, we need to calculate some more information, some more data from this 2.2 by 2 table, and in the coming up slide we will just see what do we calculate. What do we calculate is something known as 95% confidence interval. What we have calculated relative risk or odds ratio as 3.3 or 3.8 is known as point effect estimate, that you have observed, that you have got calculated from your own study. But let's say if you repeat these studies, if you or some other researchers repeat the same kind of a study following the same methodology in different parts of the world, will they get the same kind of the point effect estimate? No, they will get a variable kind of point estimate. And this 95% confidence interval tells you that your point effect estimate can vary from the lower end to the higher end. So you see there is a bold colored square and you see there is a left whisker and there is a right whisker. So these horizontal whiskers are known as the lower limit of the 95% confidence interval, which is the left end, and the higher end of the confidence interval, which is the right end. Now, three important kinds of data outcomes that you usually get is the first, where you see that the point estimate is 3.3 and the lower and the upper end is also positive and is not crossing this line of no difference. So if you see this kind of a graph, you have an x-axis and you have a y-axis. Y-axis starts from the null point, that is 1. So even if you do not intervene, even if you do not have hypertension or you do not give any kind of a drug or you give a drug, still there is some kind of morbidity in the background. So even in the control group, you can have that kind of the outcome. So even if the hypertension is not present, some patients do suffer from the cardiovascular related mortality. So this y-axis is the line of reference or the null point or the neutral point. On the left side, this favors that the control group, and on the right side, this favors the intervention group. So if you see the first point, 3.3, so that means the patients who suffer from hypertension are 3.3 times the risk of the developing cardiovascular related morbidity and mortality as compared to the patients who are not having the hypertension at baseline. And if you repeat these studies 100 times, the 95% of the times your point estimate is going to lie between 2.5 to 4.4, all in the positive. So that means clearly the hypertension poses a risk factor for cardiovascular disease in a positive manner, which ranges from 2.4 to 4.5. 2.4 to 4.4. Let's say here the second outcome. Let's say you would have got the point 1.4. So if you have got the point estimate as 1.4 and if you repeat the study 100 more times, sometimes you will not get the positive outcome and your lower end is crossing the line of no reference, and it is coming as 0.92. So that means hypertension may not be a risk factor. It is 0.92. The outcomes are less in case the patients are having hypertension, while in some of the studies the outcome is more. So this shows that your study is not powered enough, the study is not significant enough. The p-value is more than 0.05 and it is not less than 0.05 to say whether your study is significant or not significant. So to say that your study is significant or to say your study is non-significant, you need to observe the point estimate along with the 95% confidence interval. So if you have to say that your study is significant, the point estimate along with the 95% confidence interval should entirely lie on one side of the one side of the line of no reference. It can lie on the left side, it can lie on the right side to give you the significant valid estimates. This becomes all the more clear in the coming up slide. So if you see the slide, this is known as a forest plot, which is the prime outcome component of the meta-analysis. This forest plot is taken from the Tumor Biology 2014 article. This was a meta-analysis about a drug known as pioglitazone, which became infamous for its association with bladder cancer, and a lot of original studies were showing that the long-term use of pioglitazone, which is a well-known anti-diabetic drug, PPAR gamma agonist, its long-term use may be associated with a deadly outcome, which is known as bladder cancer. And finally, this study was published in 2014, and now even the updated systematic review is there in 2019. Here, if you see, originally 9 studies, for leave this overall, 9 studies starting from 2005 to 2012 have been included. Now let me try to explain this forest plot completely. You see that the first column says study ID. Study ID means the original study that has been included in the meta-analysis. So you see here the name of the author is written and the year of the publication of that study is being included. And then you have the X and the Y axis, X axis and the Y axis starting from 1. That is the null, that is the neutral point. So on the right side, it favors the intervention, that is pioglitazone is associated with bladder cancer, and if the estimates are coming on the left side, it says that the pioglitazone is not associated with the bladder cancer. So let us try to see the second study, that is from the Piscini to 2011. Now here you see that the effect estimate is 4.30, that you can very well clearly see, and the 95% confidence interval is ranging from 2.82 to 6.52. The x-axis is usually in the logarithmic value, so here you may not exactly see that it corresponds to those kind of values as it is being written. This is not a normal scale. The x-axis is usually a logarithmic scale. So you can see that the Piscini et al. Study is a significant study. Well, if you see a Dormendi et al. Study from 2005, you see that the point estimate is 2.37. But if you see the lower estimate, lower confidence interval limit 9.1, it is on the line of neutrality, that is the null hypothesis, which is 0.91. So this study is not significant study. Similarly, you see other studies by Lewis et al., Lee et al., etc., which are Zhang et al. Again, your lower end of the confidence interval is crossing the line of no significance, that is 1. So they say that these individual studies are not significant enough. But this is called like a tower of power, since you have included all the studies. So here, let's say in the Dormendi et al, only 1,000 patients were included. Other studies, some might have included 500, some might have included lakhs, etc. So all these studies will give you a bigger effect size. So after pooling all the 9 studies, the point estimate that you have got is 1.50, and it is coming out to be significant, as you see that the confidence interval is ranging from 1.14 to 1.98. So all the 3 values are on one side of the line of no difference, that is vertical line 1. So that means that this is a significant result. 1.5 times the risk, excess risk is 0.5 from the control, because 1 or 100%, if you convert it into percentage, so we say 50% increased risk of having bladder cancer if there is a long-term use of pioglitazone in the patients. Now here you see that the color of the individual study point estimates is dark green, and the color of the diamond of the pooled estimate is blank. This is just to differentiate the individual studies from the pooled effect estimate. So, and the next point is, if you see that the sizes of the various individual point estimates is also different. So this also needs to be understood that if your study is bigger, that means more sample size, more patients are included, then the size of the individual point estimate square or diameter is bigger, and is smaller, or the sample size included in the individual study is lesser, then the size of the individual box is smaller. So this is the pictorial representation of the numbers which are given here. So these are the individual effect estimates along with the 95% confidence interval, and 1.50 is the meta-analytic approach giving you the pooled effect estimate. Now here you need to understand that this is not average. This is not like N1 plus N2 plus N3 divided by N. No, this is not like that. This is not average. This is your weighted pooled average, where you give different weights. Now here, please try to see that the total is always 100, whether you include 9 or whether you include 90 studies. But each individual studies, as we read in the initial slides also, is not given the equal importance. Some of the importance which is equal to the weight that is being given to each study. Now here you need to understand the correlation of this weights which is being ascribed to each individual study. Some of the weights are very less, like if you see Song et al study which is being given only 1.61, while some of the studies are being given very high weight, like if you see Newman et al 2012 study in which the weight given is 16.96. So how does these weights are being ascribed? Actually you do not ascribe, this automatically comes in the software, but what is the basis? There are a few correlations that you need to actually appreciate here. If you see that the weight is high, let's say in the Newman et al study, if the weight is high, the confidence interval is very small, very narrow. See here, the point estimate is 1.22 and the 95% confidence interval is very narrow, ranging from 1.06 to 1.43. Contrast to this Newman et al study, if you see the Song et al study, which has been given the minimum weight of 1.61, you see that the point estimate is 2.09, while the interval is very wide, and it is even crossing the line of no difference, ranging from 0.26 to 16.8. So there is an inverse correlation between the weight of the study and the width of the confidence interval. More is the width of the confidence interval, lesser is the weight given to each study. So this is the correlation I want you to appreciate in this slide. But what is the basis of giving this weight to each study, you see that the weight being given is different for each study. There are two important bases which are interrelated. The first important point that decides the weight which is being given to each study is the total sample size. That is the total number of patients that are being included in each study. So just by seeing the weights, you can understand that the maximum number of patients are being included in the Newman et al. Study, which is being followed by the Lewis et al. Study, and then which is being followed by the Shang et al. Study. And the second important component which decides the weight being given to each study is the number of events, number of patients who suffered, who observed the outcome of interest in each study. More is the number of outcomes, more is the weight being given to the study. So here the number of events or the outcome rate is higher in case of Newman et al. Study, while the number of patients, that is number of bladder cancer cases, were very small, very minimal in case of Song et al. Study. This is known as a forest plot, which describes the individual studies along with the pooled generated effect estimate. So you need to clearly understand that this is not an average, which may give you 1.82. Average means you give equal weight to each study. While here some of the studies are bigger, larger, which we have recruited many more number of patients as compared to the smaller conducted studies. So here we calculate is known as weighted pooled estimate, weighted pooled average, which has come out to be 1.50 and has been given different weights to each different study. There are few another points that has been described, which we will see in the coming up slides. That is the heterogeneity measured, which are being calculated as I-square and the p-values. And here another thing that you see is the random effect analysis. What is that? Let us see in the coming up slide. So test for heterogeneity. What is heterogeneity? Heterogeneity is nothing but how heterogeneous the results are, that we just saw, that what is the variance that we have got. Some of the results have given a higher effect estimate. Some of the studies, original studies, gives very small effect estimates. Some of the studies are showing negative correlation. Some of the studies are showing positive correlation. So depending upon how heterogeneous the individual studies that are being included in your systematic review, you have the heterogeneity. Doing the heterogeneity test is important to understand which statistical model you are going to use to perform your meta-analysis process. So for this, you use two important tests. One is your Cochrane Q test and another is your I-square test. The Cochrane Q test gives you a p-value, while the I-square test gives you the values in the percentages. This is quite opposite. Please try to understand. If the p-value is small from the Cochrane Q test, that is less than 0.1, 0.01, the variations are not due to chance, and you say that the methodological heterogeneity is present. While a large p-value, that means heterogeneity test is non-significant and the variation is considered to be just random or due to chance, and you say that the heterogeneity is not present. Similarly, in the I-square test, which gives exactly the opposite result, if the value is large, more than 50%, you say the heterogeneity is present, while a small value, less than 50%, you say the heterogeneity is not present. This is exactly opposite. Smaller p-value, heterogeneity is present. Smaller I-square value, no heterogeneity is present. So one needs to understand. And what does this heterogeneity test gives you? This tells you that if the results are heterogeneous, you are going to use random effects model, which is known as Darcer-Mounian-Lade method. And if the heterogeneity results are negative, you say that the individual studies are not very heterogeneous. You use fixed effects model. So just to show you that in the pioglitazone forest plot, we had used, the authors had used random effects model because the individual studies were quite heterogeneous. You are going not going to actually perform this analysis yourself unless you are a statistician. We have a lot of softwares which readily suggests you which model to be used and which readily gives you the tests of heterogeneity, ascribes the weights to the individual studies, and also suggests you which model among fixed and random to be used in cases of the meta-analysis to be performed in case of your research question. The next important thing is the publication bias. Again, the publication bias, there are two things. You get a graph, you get a value. So publication bias is that you expect that if there is a research question, you are bound to get the results right from 0 to the maximum value. So you can get the negative results along with the positive results. So we say that the publication bias is present only if the positive results or only the negative results are present. And we say the publication bias is not present if the results are uniformly published. Usually in cases of intervention, what we see is that we only get significant publications and the negative results are selectively not published or they go into non-publication. So again, we perform something known as publication bias test, and here if we get the p-value more than 0.10, we say that the publication bias is not present, and if the p-value comes out to be less than 0.1, we say the publication bias is present. And this test is actually known as Eggers and Begg's test. Okay, so this is the graph, this is the inverted funnel plot, this is what we get. If the publication bias is not present, you can see that the results, these actually the circles, they say that a whole over some of the negative results along with some of the positive results are present. And we say if only we get the studies here in the right corner of this funnel, in inverted funnel, we say that the publication bias is present and selectively the negative results are being not published. So basically these plots the effect sizes against the sample sizes of the participant that are being included in individual studies. So next component is known as the test for interaction. This is not mandatory for all the meta-analysis. This is mandatory if you try to include some important subgroup analysis. For now subgroup analysis, so this is a kind of a plot that you see. This is a kind of a subgroup analysis that was being performed in one of my studies, where I tried to assess the usability of statins in case of patients with the breast cancer to improve their survival, and we tried to the subgroups also, subgroup analysis also. This was a meta-analysis of lot of this was a meta-analysis of lot of observational studies. In this observational studies, we included the cohort studies and we also included the case control studies. And we could get 24 studies at that time. This was published long back in 2012, and we got 24 studies in that, and finally we got the relative risk, pooled relative risk as 0.99, and of course you can see that the 95% confidence interval is crossing the line of no difference, that is 1.94 to 1.04. So clearly it is suggesting that statins may or may not be useful in cases of breast cancer. We cannot give the valid results from conducting the present systematic review. This is a test of heterogeneity, and if you see the p-value is very small. So if the p-value is very small, it is considered that the heterogeneity is present. And similarly, you see the I-square value is crossing the cutoff, that is 50%, again suggesting heterogeneity is present. And you see the publication bias, it is 0.71, 0.32, so it is more than 0.10. Now, since we included the two important study designs, the cohort study designs and case control study designs, we wanted to understand that if we just pull the cohort studies or if we just pull the case control studies, do we get significantly different results? This is not a simple t-test or a chi-square test. Here we perform a very different kind of analysis. This is different from calculating a simple p-value. This is known as p-interaction value, or you can also, this is like the different two groups of studies. This is two groups of studies. So if we see that the cohort, if we pool 13 cohort studies and 11 cohort studies, and if we get DP interaction value, that is 0.35. So even if we just pool the cohort studies or just pool the case control studies, again the results are not going to be significant, and we have got the p-value more than 0.05. This is an important component, p interaction, that is very important if you include different study designs or different kinds of groups in your systematic review. So now let's come on to, so we have done all the 4 steps of meta-analysis. So just to apprise you regarding the various softwares that are available for conducting the meta-analysis. Again, I always divide into freely available softwares and the paid versions. Certainly, we always go for the review manager, that is RevMan, available through Cochrane. It's a freely available software. Please don't think that only for the Cochrane systematic reviews you use this kind of a software. You can also use this software to conduct other kind of meta-analysis. Also meta-analysis, epi-meta, easy meta-analysis, meta-test, meta-stat, these are all freely available meta-analysis specific softwares that are usually enterprise version. That is, you just enter the original data from the individual studies and you get all the graphs and the results automatically. You don't have to write the programming language to get your meta-analysis results. While these are the few, that is comprehensive meta-analysis, our institute has subscription to that. MetaWin, WEZ meta-analysis, these are the paid versions, and these give very beautiful graphs, beautiful results. These also suggest you, depending upon the heterogeneity of your original studies, and does all the processes for you. But these are just the meta-analysis specific software. So if somebody wants to go deep into this kind of research studies, one can also include some general stats packages. In this, the most important nowadays, it's being used is the R. Otherwise, you can use SAS, Stata, S+, StatDirect, and there are many more that are available. Here I need to mention that GraphPad and SPSS, which are being usually used by the students to analyze their thesis results, cannot perform the meta-analysis. And the beauty of RevMan is that there is a Cochrane handbook which tells you, which guides you step by step how to use this software and how to perform the meta-analysis and systematic reviews. Okay, so this needs to be remembered again. This we have understood in great details. Prisma and MOOS, and you can get from equator.network.org all these checklists to report systematic reviews or meta-analysis pertaining to your particular question. And finally, it is just to give you a brief about what can be the types of questions. People ask me, is it only the drugs efficacy and safety? No. There can be many types of questions in the medical literature, starting from therapy, asking about the efficacy and safety of a particular intervention, drug, surgical, etc. But there can be questions regarding etiology, harm, just we saw pioglitazone in bladder cancer, some contributing factors for like hypertension, for cardiovascular disease, prognosis, the outcomes if you do not do anything, like in case of cancers. Then diagnostics, sensitivity and specificity. We had starred checklist also. You can perform the systematic review and meta-analysis for diagnostic testings also. Harm, etiology, for example, in COVID-19, you can have that etiology for the specific mutations and the lethality. Then prevention strategy, you can have like prophylaxis, you can have some kind of social intervention, you can have vaccines, some kind of lifestyle intervention for the primary, secondary prevention, like aspirin for the secondary prevention of high-risk cases in case of cardiovascular mortality. Then you can have quality improvement questions also. These are basically system questions. And nowadays you have a lot of economic decisions. You have some more specific kind of evidence generation, like Drummond's criteria for economic decisions, or health economic and outcomes research studies, or cost of illness studies. And then finally we have some prevalence studies, prevalence studies like burden of illness. What is the total burden of a particular kind of a disease or an illness in the society? You can perform systematic reviews, even meta-analysis in these kind of prevalence studies. I'll just show you how I have done the two types of meta-analysis in the red colored two different types of questions. Now see this question. This is regarding the therapy, and I showed you the search strategy also, which I have used to perform this kind of meta-analysis. This is actually a higher end of a meta-analysis. This is known as a network meta-analysis. This was done in a disease known as diabetic ketoacidosis. You know that diabetic ketoacidosis is acute, life-threatening complication which can happen in cases of type 1 diabetes mellitus patients, though some type 2 also nowadays suffering from diabetic ketoacidosis. And the primary form of treatment is the rapidly acting insulin or the regular insulins. Now the question of interest in this network meta-analysis was that in the recent past decade, around 10 to 15 years, we see that a lot of newer insulin analogs like Lispro, Glargine, etc. have come up. Glulicine, Aspart, etc. have come up to treat the patients of DKA instead of the regular insulin. Now, insulin is insulin. So what do we wanted to understand? We wanted to see whether these newer insulin analogs, they pose any kind of a superiority with respect to the efficacy or the safety outcomes in cases of diabetic ketoacidosis. Just to summarize the steps that we have understood, in the previous slides, we have used library clinicaltrials.gov. Again, to revise, PubMed is freely available, Cochrane Library is freely available, clinicaltrials.gov is freely available. Embase and Scopus are the paid version. And in this, we collaborated with PGIMER Chandigarh, who have got the subscription for these two databases like Embase and Scopus. So this was the databases, and if I have just cut short the methodology, but if you happen to read this whole paper, you will see that there was a stringent inclusion and exclusion criteria, and the research design that I was interested was only randomized controlled clinical trials, and we wanted to evaluate, compare the relative efficacy and safety of insulin analogs for the management of DKA, diabetic ketoacidosis, using indirect treatment comparisons, which is known as Bayesian network meta-analysis. And finally, we got 10 RCTs which were randomized. The total number of patients that were included in this meta-analysis were 435. Total number of interventions that were included in this systematic review was 5. Lispro, Glargine, regular insulin, Glulicine, Aspart, and regular insulin. Both efficacy and safety were compared. And what is the final conclusion that we could draw from this clinical trial, this systematic review, was that newer insulin analogs, they were found to be equally effective and safe as regular insulin. While you know that the newer insulin analogs are much more costly as compared to the regular insulin, which is available in the market through decades. So to actually incorporate newer insulin analogs in the treatment of DKA in a cost constrained settings like in India, we need to perform cost effective analysis. Why? Because insulin analogs can be given in a non-ICU setting, while regular insulins needs to be given only in the ICU setting. So the cost-effective analysis of the newer insulin analogs is required before these expensive agents can be used instead of using regular insulins. I have not given you actually the numbers, the differences, the point estimates, just to keep this question easy to understand for you. So this was one therapy question which was analyzed in a meta-analytic manner. So let's see another type of question which is known as a contributing factor. Here I wanted to understand that there is a very important disease nowadays prevalent in the society, and it has actually increased in the era of COVID-19 because of the long-term use of computers and the sitting positions and all the work being done through home online mode, the low back pain. And we wanted to understand that vitamin D, which is a very important component in maintaining the musculoskeletal health, thus the decreased levels of vitamin D, is it inversely correlated with the low back pain? So as the deficiency of vitamin D increases, the back pain prevalence increases. This was the major hypothesis in this kind of a meta-analysis. So you can understand that you will not generate hypovitaminosis D in patients. So what kind of studies that you are going to get? You are going to get only the observational studies. So you are going to get only the observational studies, including case control studies, cohort studies, and the cross-sectional studies. So we have pooled all these observational studies in this meta-analysis. And if we see finally, we got 14 studies in which 6 studies were case control studies, 6 studies were cross-sectional studies, and 2 studies were cohort studies. The total number of patients that were included in this meta-analysis was 2,602. And we have got the pooled value as 0.72, ranging from 0.60 to 0.83. So they say as the 72% higher chance of having the low back pain disease, and the confidence interval is also not crossing the line of no difference. So the results are quite significant. And they say that 72% chance or higher chance of getting low back pain if the person is having hypovitaminosis D or the deficiency of vitamin D hormone. Now, and a few other points that you can see is that marked heterogeneity was observed. So if the heterogeneity was marked, we have used random effects model, and WPPR is the weighted pooled prevalence rate along with the 95% confidence interval that has been presented in this kind of a meta-analysis. So this was to brief you that how different questions can be answered through meta-analysis approach. And finally, to apprise you of the various resources that you can go through to dig deep into understanding the conduction, the science behind the meta-analysis and the systematic review is the first. Definitely the Bible is the Cochrane Handbook for Systematic Reviews of Interventions. This is freely available in the PDF form. Please visit the website also. Please visit this website. Now this website gives you the handbook. This website gives you the ROB 2.0 tool, Ruby's tool, Robin's tool. This website gives you the RevMan. This website also gives you a lot of information regarding the free training sessions for different modules. There are a lot of free training sessions are being happened being conducted by the Cochrane Training Center from time to time. You can keep on visiting this site and can see when these training sessions are being available. Apart from that, you can also go to the Cochrane Colloquium. This is a huge conference conducted every 2 years, and this was to be conducted this year, but because of the COVID-19 has been postponed, and now it is going to be conducted in Toronto in 2022. And you can actually present an abstract also, but that has to be very much novel. That means you need to give something new regarding methodology, about the meta-analysis, and which can be of interest to the high-end researchers. And finally, you can go to the CMC Vellore site. And this was actually the South Asian region, and now the Cochrane Colloquium is going to have the center or South Asian region center in this BBMC center, Professor B.B. Moose Center for the Evidence Informed Healthcare and Health Policy. This centers, which is present in the CMC Vellore, Christian Medical College Vellore, they keep on conducting a lot of workshops, lot of trainings, which are paid as well as free from time to time regarding conduction and the reporting of systematic reviews as well as the meta-analysis. So all the students who are interested in this kind of research can visit various websites and can update themselves from time to time about from these various web resources. And thank you very much. And you can contact me for any further queries through this website, and I'm also open for any queries right now also. Also,
Hello, ma'am. My voice is audible?
yes, of course you are audible. Hello, yes,
Thanks
I
a
am.
lot, ma'am.
Yes, you are audible.
Yes, ma'am. Ma'am, thanks a lot, ma'am. Very informative session and excellent session. You gave a brief about the overviews of systematic reviews and meta-analysis. Our students and participants are very wonderful session. Gives the comments. Thanks, ma'am. Now I would like to invite Dr. Jaspreet,
Also,
ma'am, for vote of thanks. चलेगा.
Thank you, ma'am, for this wonderful and informative lecture. Really, it would be helpful for evidence-based research, especially your evidence-based pyramid and various types of research what you have shown. Thank you so much, ma'am. I would
my
like to
pleasure, my
express our
pleasure. Thank
sincere
you.
thanks. I would like to express our sincere thanks to both the speakers for sparing their valuable time for us despite their busy schedules and widen our knowledge spectrum with an excellent coverage. With this, we finish session for the first day. Have a nice day and we'll meet tomorrow. Thank you so much.
Thank
Thank